Hi, everyone. Welcome back to day 2 of this amazing conference. Um, so please grab a seat, and we'll have today's session started. Um, so many years ago, everybody in human genetics was doing linkage analysis using family data. And then in 1995, Neil Risch and Kathleen Mary Kangas published 2 pages in Science using simulations to convince everybody in the field that you guys are just wasting money. If you keep doing linkage analysis on complex traits, the statistical power is just not going to be good enough. And since then, everybody is sort of switched. Very few people are still working on linkage analysis for complex traits, complex diseases. And then, of course, genotyping and sequencing have become so much cheaper. These days, we're doing genome-wide version of association analysis for pretty much any phenotype you can think of. So we have left that family analysis behind. The entire field has left it behind. But then in more recent years, the field realized that we're probably a bit naive to completely abandon the older strategies. We realized that genetic association study based on independent people can actually give you confounded associations, plagued by a variety of technical issues that we have a better understanding these days. So the whole field is now revisiting how to leverage family data more efficiently and effectively in today's complex trait genetic studies. So this is a session about exactly that. We have 3 excellent talks. The most impressive thing about these 3 talks is that they are essentially all led by undergrad trainees. And it's also a revisit of 3 presentations we have seen in the previous year. This is going to be an update, a more complete version of all 3 projects. First presenter is going to be Matthew Howell from UCLA, and second is Dhruva Jaishankar from also UCLA. I'm the 3rd presenter, but I'm presenting on behalf of Qinwen Zheng, who's our undergraduate student who has recently graduated. I actually saw him yesterday, so probably he should be here. But I was assuming that because he had recently graduated from college, he will be on a beach somewhere. So that's why I'm presenting on his behalf. So without further ado, I'll let Matthew take the podium. Yeah.
All right, thank you, Q, and it's nice to see everyone again. So I'm going to be revisiting a presentation that I gave last year about my project, Assortative Mating Across Time, or AMAT. And big thanks to everyone at SSGAC, including Patrick, my mentor, Dan, John, and everyone else at SSGAC and everyone who's collaborated on this project. All right, so first of all, assortative mating is when parents are more similar phenotypically or genetically than if pairing were random. So let's say that row 1 and row 2 are meant to be paired to be parents, and they vary in terms of educational attainment. So row 1 has high educational attainment, last row has low educational attainment, etc. And when we have assortative mating, we would expect it to look like this, where people with higher educational attainment are also pairing with people who have high educational attainment, and vice versa. All right, so assortative mating can be measured with phenotypes, and that's what we call phenotypic sorting. But it can also be measured with PGIs, and that's what we call genetic sorting. And AMAT will estimate the trajectory of genetic sorting over time using the PGIs of only the children. So no data on parents are required, which is very exciting. And AMAT can help us explain several things, including changes in disease prevalence and severity, as well as social phenomena such as changing inequality and social mobility. Let's talk about the method for a little bit. So assortative mating across time will measure the level of genetic sorting, which we're going to call M of T, simply as the mate PGI correlation at time t. So how similar parents' PGIs are at time t. And time t is just going to be when their child is born. So assortative mating will introduce both within and cross-chromosome linkage disequilibrium, or what we call LD. And we can estimate assortative mating by measuring the level of LD at a very high level. However, within chromosome LD, in large part, is not due to assortative mating. However, cross-chromosome linkage disequilibrium does not face that same challenge, and cross-chromosome allele pairs actually vastly outnumber within chromosome pairs. And so if we compute the contribution of cross-chromosome allele pairs to total PGI variance, that gives us an estimate of the degree of cross-chromosome LD, and that will give us a close approximation of the degree of assortative mating. All right, so we're going to use data from two different data sets here, both UK Biobank and All of Us. The story is similar with both of them. They're restricted to participants of European genetic ancestry and residualized on the first several principal components of the genetic data to control for population stratification. For UK Biobank, we use data from partition 3, which has a sample size of 150,000. And here we apply both family-based and population-based PGIs. Population-based PGIs are made with PGI repo weights, and family-based PGIs are made with Tan et al. Weights. Similar story with All of Us, however, our sample size is larger here with 210,000 participants. We actually increased the sample size recently, so we made use of that. And we only apply family-based PGIs in All of Us due to data use restrictions, and those are based on the same Tan et al. Weights. All right, so let's talk about what we have changed since last time. And we have extended the Amat theory in multiple exciting ways, and the first is we now measure cross-trait assortative mating. And cross-trait assortative mating can be described as so. So let's say that we are pairing the same people with each other, but this time row 1 varies in terms of educational attainment, and row 2 varies in terms of self-reported health. If we have cross-trait assortative mating between these two groups, we might see something like this, where people who have high educational attainment are pairing with people who have high self-reported health, and vice versa. We're going to be measuring cross-trait assortative mating using that same mate PGI correlation at the time their child was born. All right, so let's talk about some results that we have for cross-trait assortative mating. This is a selected result, but we have many, many more if you're interested in looking at that. And this is going to be cross-trait assortative mating for self-reported health and educational attainment, so just like our example there. And I'm going to guide you through these axes. Yeah, this works. Okay, so first of all, we notice that we have assortative mating here on the y-axis. And that's just going to be our measure of genetic sorting, which is the mate PGI correlation at the time their children was born. So we're inferring that through our method. And here is just going to be that birth date of the child. And as we can see, we have a pretty nice increasing trend for mate PGI correlation between self-reported health and educational attainment. And we quantify that increase by running a linear regression through our assortative mating estimates, which are right here in dark blue. And then we use the bootstrap to get a standard error on those linear regression slope estimates. And if you look up here in the top of this legend, we see that our slope is positive and highly statistically significant, which means that we do say that assortative mating is increasing over time, cross-trait between self-reported health and educational attainment. So the mate PGI correlation is increasing over time. Also, we have a measure of mean assortative mating, and also we see that that is positive and statistically significant, which means on average, mate PGI correlation is also positive and statistically significant over this period. And I should say, I forgot to say that we are using population-based PGIs in the UK Biobank. The colors refer to different data sets, so blue is from UK Biobank, All of Us is in red, and we are using population-based PGIs to generate these results here. So now let's look at family-based results. And as you can see, we now have results in red, which means that we have data from All of Us. And as we can see here, the only significant result that we get is for mean assortative mating in All of Us. So again, we detect positive and statistically significant mean assortative mating, mean mate PGI correlation over time. And we also notice that our family-based results are attenuated compared to our population-based results. And we say that these results are attenuated likely because the family-based PGIs are based on a smaller sample. All right, so now let's talk about the second element of our extended theory. So first of all, we estimated cross-trait assortative mating, and now we also estimate phenotypic sorting over time. And our measure of phenotypic sorting is just going to be the correlation between mate phenotypes, so analogous to our measure of genetic sorting, which is M of t. Now it's just going to be the correlation of mate phenotypes at time t. All right, and I'm going to guide you through exactly why we decided to measure phenotypic sorting. So we're going to assume that mates sort only on phenotypes and not PGIs. So then say that we observe changing genetic sorting, which is the measure that we were looking at before, but constant phenotypic sorting. We say that changing PGI predictive power might explain this discrepancy. And phenotypic sorting will help us evaluate whether changing PGI predictive power can explain the changes that we see in our genetic sorting estimates that we were looking at before. So this is going to be our measure of phenotypic sorting, and it's going to be proportional to our genetic sorting estimate, M of t, which is right up there, divided by our PGI predictive power at time t squared. That's going to be R of t right there. Notice that our measure of, or our estimate of phenotypic sorting is only proportional to this ratio. So technically, it's not a valid estimate of the level of phenotypic sorting at time t. However, because it is proportional, we can use the slope on our estimate of P of t to test whether the level of phenotypic sorting is constant or changing over time, which is exactly what we want. All right, so I'm going to give you guys an example of what our phenotypic sorting estimates look like. And for the purposes of this presentation, I want you guys to only focus on the slope estimates in this legend here. So as we can see, we are now looking at within trait assortative mating within trait assortative mating for BMI. So it's not cross-trait assortative mating between two different phenotypes. We're just looking at genetic sorting for one phenotype alone. And first of all, again, these axes are going to be analogous to the ones that we were looking at before, where the y-axis is the mate PGI correlation, and the x-axis is going to be the year of birth for our samples. And as we can see, for BMI, using population-based PGIs in the UK Biobank, we see that the slope is not statistically significant, which means that we say that assortative mating, genetic sorting, is constant at this time, or across all of these years. And now let's introduce our measure of PGI predictive power, R of t. So our PGI predictive power is going to be on the y-axis here, and this is going to be the predictive power for our population-based PGIs that we use to generate these genetic sorting results. And as we can see, that also looks constant. And so when we generate our phenotypic sorting estimates by dividing this measure of genetic sorting right here by our PGI predictive power right here, it's unsurprising that also our slope on our phenotypic sorting estimates is also constant. However, we have a slightly different story when we look at our estimates using family-based PGIs. Particularly if we look right here, we have a significant slope. So we say that within trait assortative mating for BMI, when we apply these family-based PGIs in the all of us data set, this red trend line right here, we're saying that that level of genetic sorting is increasing over time. However, let's look at our PGI predictive power for those same PGIs that we use to generate those genetic sorting estimates. We see that it looks like it's increasing over time, right? And so when we divide these genetic sorting estimates, again, by the PGI predictive power here, and we get a slope on those estimates for phenotypic sorting, we see that it is no longer statistically significant at the 0.05 level. And so we're observing constant phenotypic sorting, but a changing genetic sorting. And so we say that this increase that we're seeing in PGI predictive power over time might explain that discrepancy. So now I'm going to give a quick rundown of the general results that we've produced. First of all, we're going to look at within trait assortative mating. For all of these phenotypes that we see here, a wide range of phenotypes, but we have more that don't intersect across different types of PGIs that we are using across data sets. So a darker shade of green in this figure right here means that we're observing more significant results across the different types of PGIs that we applied. And again, those are population-based PGIs in UK Biobank, family-based PGIs in UK Biobank, and also family-based PGIs in all of us. And as we can see, first of all, we're going to look at mean genetic sorting, or mean assortative mating that we were looking at in those plots before. And as we can see, across all of these traits, we do observe mean genetic sorting for most of these traits, save for a couple. Especially for educational attainment, ever smoker, and height, we have positive mean assortative mating, or yeah, positive mean genetic sorting across all of these different types of PGIs that we're looking at. Now let's look at changes in genetic sorting, and this is going to be that slope on genetic sorting that we were talking about before. As we can see, we don't observe as many significant results when we look at the slope on genetic sorting. However, we do see for traits like BMI, educational attainment, ever smoker, we are picking up some signals of changing genetic sorting over time. And then when we then measure phenotypic sorting, which we're using to compare to our statistically significant genetic sorting estimates, we see that a lot of the time our significant genetic sorting estimates are no longer significant when we estimate phenotypic sorting. So for BMI, for example, we observe statistically significant genetic sorting, but then is no longer statistically significant, that slope on our assortative mating estimates when we measure phenotypic sorting. However, we do see that that slope is preserved for educational attainment and self-rated health. That's specifically in the All of Us data set, if you're interested. And so we say that because we observe statistically significant genetics or changes in genetic sorting, and also statistically significant changes in phenotypic sorting over time for these two different phenotypes, we say that changing PGI predictive power over time cannot explain all of the changes that we're observing in our genetic sorting estimates. All right, so now we're going to look at cross-trait assortative mating between educational attainment and the phenotype that you see here in this first column. Again, darker green just means that we are observing more significant results. And again, we see widespread average genetic sorting across all of these traits, which is very, very interesting. And then when we move to measure the changes in genetic sorting over time, we do again observe for several of these traits at least one significant slope on our genetic sorting, including for BMI and self-rated health, as well as subjective well-being. Now, when we adjust for changing PGI predictive power, we see that we maintain those statistically significant slopes a lot of the time across these different phenotypes. This one's a little bit of an odd one out because it's only marginally significant. It probably wouldn't pass multiple testing corrections, but we see that for BMI, depression, as well as self-rated health, the slopes on our phenotypic sorting estimates are also statistically significant, which again means that changing PGI predictive power cannot explain all of the changes that we're seeing in genetic sorting. All right, so just to wrap up, in AMOT, we now measure cross-trait assortative mating and also phenotypic sorting. And the main takeaways that we have here is that we find widespread cross-trait assortative mating with educational attainment across the different PGI types and data sets that we apply. And also, changing PGI predictive power may explain some of the changes that we are seeing in genetic sorting for some phenotypes, but not all of them. And that's it, and I just wanted to thank everyone at SSGAC again, as well as all of the collaborators on this project. And yeah, thank you so much.
We definitely have some time for questions.
Hi.
Thank you. Fascinating presentation. I think I understood some of it. I had a couple of questions, but the first one I wanted to follow up on was just what time means in your analysis. So,
Yep.
As I understand it, you're comparing birth cohorts, right? That it's the time dimension is the date of birth of the person whose genetics or phenotype you're measuring.
Yep.
So that tracks for me when you talk about changes in genetic sorting over time. Genetics are fixed; they don't change across the life course. Phenotypes, not so much. So when you talk about changing sort of PGI power or PGI predictiveness over time, what you mean is differences in the R-squared for a PGI for different birth cohorts, which also means, I think, in the context of all of us in UKB, you're describing differences between people of different ages. And that seems to have a qualitatively different meaning than what you're doing with time on the genetic dimension. So you're not necessarily observing changes over time, you're observing changes across the age span. And I'm curious what the implications of that are for the adjustment part of your model, where you're now correcting your estimate of the slope in genetic correlation between mates for the trait or the PGI trait correlation for those birth cohorts. Do I have that right?
Yeah, I think I understand your question. So let me go back to look at our phenotypic sorting estimates, just so we have a good look at that. There we go. So you're right that our x-axis on our genetic sorting estimates is just going to be the time at which our samples are born, because we're using children to estimate the trajectory of assortative mating over time, which is the parents' PGI correlation, right? So PGI predictive power at a specific point in time is just going to be the correlation between the samples' PGI with their actual phenotype. And that does change over time, right? So we're...
But you don't have time here. You don't have time here, you just have age.
Right, so yeah, so at their specific birth year, that is when we're plotting PGI predictive power. So I understand what you mean when the interpretation is a little bit different.
I mean, it's just a totally different thing, basically.
Right,
Like,
yeah.
you know, when Maria Gleamore did this in HRS, and I guess, oh, what was her name? Somebody did it first in the Fells longitudinal study, looking at BMI correlations across birth cohorts. There was this observation that PGI power seems to rise. But in those cases, they had longitudinal data, so they were able to observe different birth cohorts at the same ages to have a constant sort of, like, this is BMI at age 40 for people born in 1940, 1950, 1960.
Yeah.
And you don't have that here, and I think that's really consequential, certainly for BMI, I suspect for self-rated health, perhaps for some of the other traits as well, because the age of the GWAS sample, you know, is going to condition the predictiveness of the metric for the, you know, the target population. If people are 80 versus if they're 25, it's going to be really different.
Yes.
And then I think, you know, it's worse than that because of the sample selection into all of us, and UKB is going to be different for people of different ages. So I think it really, like the first part of this, I think I totally follow, but I'm curious what the potential implications of that slippage are for this adjustment that you're making. Because I think that's sort of the crux of it, right? You're saying we want to evaluate these changing genetic assortments over time against a background of something that the PGI may be less good in these earlier born cohorts, and that's why it looks like assortative mating is weaker. I'm not sure that's. I guess I'm not sure you can do that.
That's a great point. Yeah, and I haven't fully considered that, but thank you so much for talking about that. That's something that I'll definitely want to look into. All right. Are there
Because of time, we actually have to move on to the next speaker, but I see several other people raised hands, so you can definitely follow up with Matthew after the session.
Cool. Thank you so much.
Next presenter is Juba.
Okay, cool. So yeah, hi everyone. Today I'll be talking about how genetic relatedness matched association studies can increase population coverage, reduce bias, and increase power relative to traditional GWAS. So I'll be talking about what a GWAS is, what a grandma study is, and how it works, and some empirical results that we have. So, you know, in a GWAS, we want to find an association between genotype alleles and phenotypes of interest. But one of the big problems with traditional GWAS is bias from population stratification, where we have systematically different allele frequencies in different subpopulations, along with systematically different phenotypes. And so the way that researchers typically combat this is by subsetting to continental ancestry samples, typically the European subsample, but this restricts genetic diversity, and other forms of bias can still creep into the estimates. And so, another way that researchers try to combat this is through sibling GWAS. And so, conditional on siblingship, genotypes are randomly assigned, and so you end up with unbiased estimates. But because you're only using siblings, you end up with much smaller samples with low power. And so GRAMMA is the method we propose to overcome the shortcomings of traditional GWAS. And so similar to how a population GWAS finds the effect of an additional allele relative to the population mean, and sibling GWAS finds the effect of an additional allele relative to the sibling mean, GRAMMA makes these comparisons relative to a relatedness group mean, where a relatedness group is defined by grouping individuals who are at least as close genetically as some threshold value of kinship specified by the researcher. And so by allowing this threshold value of kinship to vary, GRAMMA becomes a generalized model of sibling and population GWAS. So here's a quick visualization to help show you how GRAMMA works. So consider the entire population, where individuals who are closer together are more closely genetically related and have a more recent common ancestor, and so have more similar allele frequencies. Traditional GWAS only uses this smaller, continental ancestry subset of the data. But with GRAMMA, you could imagine setting a kinship value at the threshold corresponding to siblings. So GRAMMA will group people according to their siblings in the sample, take within group effects, and average them out over the population. And so here, individuals without siblings are dropped, and so this is equivalent to a sibling GWAS. And so you end up with unbiased estimates, but again, because you're only using siblings, you have this small sample size and low power. But, with GRAMMA, you could relax the relatedness threshold, for example, to the level of third-degree relations, first cousins, for example. So as you can see, you have more relatedness groups and larger relatedness groups. And so although some bias is introduced into the estimates, you also end up with these larger sample sizes, and so you have more power. And so that really shows you the advantages of GRAMMA. There's no need to limit the sample, and so you can use more individuals, including those from underrepresented ancestries and samples outside of ancestral subgroups that typically aren't used in traditional GWAS. And so all of this leads to increased power to estimate direct effects. And so from that visualization, we also see this kind of bias variance trade-off. At relatedness criteria, such as siblings, we typically have estimates with low bias, but also lower power because of the small sample size. And as we relax this relatedness threshold to third and fourth degree relations, we increase individuals, leading to more power, but also introduce some bias into the estimates. And so GRAMMA can actually get a better bias variance trade-off than that from population GWAS, and this is because by holding some amount of bias fixed, you can reduce the sampling variance by using more of the sample. And so to quantify this trade-off, we've developed this kind of trade-off statistic that's a weighted average of the variance of the estimates and the bias in the estimates. And so a researcher would, theta is chosen according to researchers' preferences, and they would want to choose the degree that has the minimum t of theta. And so when theta is equal to 1/2, this corresponds to minimizing the mean squared error of the estimates. So we have some empirical results having applied GRAMMA to UKB data using multiple ancestries. We ran King on the UKB data to get relationships between individuals. And we ran GRAMMA with 4 relatedness thresholds: siblings, first-degree relations, which includes parent-offspring relationships, second-degree and third-degree relationships. And so we did this for BMI, height, and educational attainment. And in the sample, this graph shows you the number of focal individuals that we have. And so what this means is basically at the level of siblings, we have about 41,000 people in the sample who have a sibling, and at the level of third-degree relationships, there are about 140,000 people who have someone who's at least as close as a third degree relation. So the first result we can look at is power. And so what we have here is a Miami plot with SNPs on the x-axis and minus log 10 of the p-value on the y-axis. And so, and along with, the degree 3 estimates and the siblings estimates. And so what you see is as we move from the siblings estimates to the degree 3 estimates, we end up getting a lot more genome-wide significant associations, especially around chromosomes 16 to 18. We see a similar trend in height with far more significant hits at the third degree level. For educational attainment, although there are no significant hits, we do see that at the third degree level around chromosome 11, there are more p-values that are closer to this genome-wide significance threshold. And as GRAMMA is applied to larger and larger samples, we'll have more and more power to estimate direct effects. And so we can also think about, this bias variance trade-off using GRAMMA. And so we ran a kind of traditional population GWAS to compare to the GRAMMA results. And so we did this using singletons in the UKB of white British ancestry, using PCs as covariates. And so what we have here is a graph of the variance of the bias of the results for the 3 phenotypes at the level of siblings, third degree relations, and in the comparison GWAS. And this bias is measured relative to the siblings estimates. So what we see is at the third degree thresholds, we have estimates that are not statistically different from 0. But, in the comparison GWAS we do. And so although we allow for, although there is the potential for bias and we allow for different true levels of bias in the different samples corresponding to the different relatedness thresholds, in practice we find that, we have these precise estimates that are not statistically different from the siblings level of bias. We can also look at the sampling variance based on the thresholds. And so what we have here is a graph of the sampling variance for the 3 phenotypes at the level of siblings and third degree relations. And for all 3 phenotypes, we see this significant decrease in the sampling variance as we relax the relatedness threshold. And so given these large decreases in sampling variance and kind of lack of increase in bias as we move as we relax the relatedness threshold, a natural question to ask is when would a researcher prefer the siblings estimates over the degree 3 estimates? And so to answer this question, we plug the values into this trade-off statistic using the best possible estimates at the lower end of the confidence interval for the siblings and the worst possible estimates for the degree 3 GRAMMA results. And what we found is that, to prefer the siblings estimates to the degree 3 estimates, researchers would have to place over 95% of their weightage on having low bias. And so this is a pretty kind of striking showcase of the advantage that GRAMMA can provide, because for most preference parameters chosen by the researcher, the degree 3 estimates are better than the siblings ones. And so we also compared the sampling variance of the GRAMMA estimates to that from SNIPAR, which is an imputation software that can run family GWAS. And so even in comparison to the unified estimator, which kind of meta-analyzes family GWAS with population effects and uses more individuals than we do in the degree 3 sample, we're able to get a lower sampling variance. So given these increases in precision and power, we wanted to see how PGIs would perform constructed using GRAMMA summary statistics. And so we did this using the All of Us data set. We ran King in the All of Us data set to get, to find 13,000 siblings, and produced PGIs using GRAMMA summary statistics at the different relatedness thresholds using the All of Us genotypes. And we ran this regression of the all of us phenotype on the PGIs, and the beta from that regression is plotted here for the 3 phenotypes, where we have the siblings and degree 3 PGIs. And again, kind of supporting the results from before, we see this increase in predictive power as we move out from the siblings threshold to the degree 3 threshold. And so yeah, to conclude, GRAMMA kind of generalizes these population and family-based GWAS approaches. And by using these extended families, we're finding huge increases in power and predictive power with no evidence of bias. And yeah, in terms of future work, we're going to be kind of finalizing the manuscript and applying GRAMMA to more phenotypes and data. And yeah, of course, huge thanks to Patrick and David for being excellent mentors, John for his coding expertise and detailed feedback, and the SSGAC, especially Dan and Alex, for helpful discussions, and past conference participants. And yeah, thanks for listening.
Questions?
Very nice talk, and this may be a pretty trivial question. So we know that the estimates of the kinship or relatedness, it's a continuous measure. So how sensitive it is to misspecification of the relatedness groups, and do you have any thoughts on directly incorporating the estimate and its uncertainty into your analysis?
Well, King is able to kind of reliably estimate the degree of relation up to the 3rd degree, and so
yeah, I guess my question is more about the individuals with a, for example, an estimate of 0.2 rather than 0.25 or 0.125. So some values in the middle. So how would you handle those?
Well, it has. Yeah, I don't know how sensitive those results are. How sensitive the King results are to those kinds of small changes in kinship. They have, intervals for both kinship and IBD, and based on those intervals, that's how they classify individuals as based on those like pairwise coefficients into different degrees of relation. Yeah, it seems like it's pretty reliable, so there probably isn't that much of a specification error problem. But yeah, that's kind of a King specific.
Okay, thank you.
Yeah.
Great presentation. So my question is like in sibling design, so we control for the kind of not only the genetic similarity, but kind of the shared environmental effects, maybe due to genetic nurturing or like pure environmental effects. I'm just curious, like in this grandmaster, so like what's our like control for kind of the shared environmental effects as compared to the kind of sibling designs, if this method kind of uses a less restrictive control on those methods, if that's the kind of one reason that results in the increase in the statistical power. So just want to hear your thoughts on that.
So we don't have any, I guess, specific controls for environment. I mean, we demean genotypes and phenotypes based on the sibling relationship groups.
I mean, like compared to say on the genomic method using general populations, if you use a sibling design, you control for the genetic similarities.
Mm-hmm.
At the same time, you control for the kind of shared environmental component at the same time, and resulting in reduction in the statistical power. In our method is kind of in the middle, right, of the sibling design compared to the using a general population.
Mm-hmm.
So like, I'm curious how this kind of shared environmental components is handled in this.
Well, so we allow for that kind of bias to be there in the, in like the degree 3 estimates, but from that kind of bias graph, we basically see that because these estimates of bias are made in comparison to the siblings level, we're basically seeing that there is no kind of significant increase in bias, even based on like those environmental factors. So yeah.
Let's thank our speaker again. All right. My name is Qiongshi. People call me Q. I'm a faculty member here at UW Medicine in the Department of Biostats and Medical Informatics, and I'm going to talk about partner choice. So I have 3 background slides to make sure everyone is on the same page about interesting study design, and then a brief introduction of the literature on assorted mating and partner choice. And then we're going to tell you something about a recent project we have finished. This is not even close to an important project we have worked with, but it's definitely the most fun we've ever had in any project over the years. So GWAS, incredibly straightforward study design. In a case control example, it's just genotype, maybe I'm being too loud. Genotype a lot of sequences for disease cases and do the same for healthy controls and compare them. You do regression analysis to regress phenotype on these single nucleotide polymorphism or SNPs and repeat the regression analysis across millions of variants and obtain Manhattan plot to quantify where in the genome that give you the association with the phenotype. People have been doing it for decades. We're really good at this type of study design. It gets really boring. But then in more recent years, people realized that these effect sizes can be severely biased. One example coming from the social genomics world is that, for example, can be genetic nurture. Parental genotype can be a confounder. Parental genotype cause child genotype. Parental genotype can actually indirectly influence children's outcome. So this is essentially a statistical confounder. So that's why a lot of people are doing, within family analysis to account for this type of confounding. Our contribution in this world is to do multi-generation analysis, essentially run GWAS on children's outcome and couple those GWAS with traditional GWAS to basically recover the true estimates for direct and indirect effects. But this is our first attempt at doing GWAS on family members' phenotype. This is different from typical GWAS. Usually you regress everybody's own phenotype on their genotype. This is regressing children's outcome on parents' genotype. So following up on this, Yuchang also did another analysis because we were thinking, now that you can do GWAS on children's outcome, how about doing GWAS on parents' outcome? Our initial intuition was some kids will give their parents headaches. What kind of genes in children will affect parents' of well-being? But then we don't have that kind of data. But then we realized this is actually a study design everybody else has been doing in the world of Alzheimer's genetics. Because of lack of cases in big biobanks, what everyone is doing in Alzheimer's genetics world is to use this self-reported parental health history for Alzheimer's as a proxy phenotype in GWAS. So essentially you are running GWAS on parents' outcome reported by children, but then the genetics is children's genetics. Then our contribution is recovered as pervasive biases in this type of study design. You can show that if you rely on this GWAS by proxy, you get spurious positive genetic correlation with higher cognition. It makes it look like cognition is a risk factor for dementia, which doesn't make any sense. And then we show that in genetic epidemiology, this is a major source of bias in many downstream applications. But this is a second attempt at this study design. It's GWAS on parents' outcome. Yeah, it's still GWAS on family members' outcome. So then I'll take one step back and then talk about assorted mating. We didn't expect there's a connection between these two seemingly completely different topics. But then we're very interested in assorted mating. Everybody in social genomics is interested in assorted mating in recent years. It's a big, but also somewhat limited literature. There are basically 3 types of papers, more or less. First type quantifies if there's evidence for assorted mating. Usually it's relying on polygenic score between couples and then see if they are correlated. Or it's polygenic score based on even or odd chromosome to see if they are correlated. The second study design tells you about parental generations of assorted mating. Between couples correlation tells you about current generation. But this is just evidence, if there is correlation or not. Another type of papers coming from mostly Scandinavian countries, they use in-laws, use co-in-laws, use extended family member to help you quantify if this kind of assorted mating has reached some kind of equilibrium in the society, or is it still continuing at a certain pace. And the third type of paper shows that if you ignore assorted mating in complex trait genetic studies, you get biased estimates for heritability, you get biased genetic correlation estimates. So it's pointing out issues if you ignore assorted mating, but then most of these papers do not tell you how to solve the problem. So this is the nature of the literature. So we're very interested in this topic. We thought it's impossible to solve, but actually it's last year at this conference we had some joint conversations with economists, just like the dinner we had yesterday. We realized that people actually study this in econ. And so this motivated us to actually read some papers in economics. The first time ever we read papers from econ. This is one example. This paper led by Pierre-Andre Chiappori. And people actually model the dynamics of partner choice. The idea, the vanilla model is incredibly straightforward, but also somewhat limited. But I wouldn't get into all the details, but the intuition is clear. So you have probably 150 different phenotypes, very complex, that drives decisions about partner choice. But their model is all these different phenotypes goes into an index value, which they call attractiveness in the marriage market. It's not physical attractiveness, has a lot of social economic stuff in it, but people sort based on attractiveness. Two people having the same value are essentially equivalent in the marriage market. But the reason why these two people have the same value can be completely different, can be based on very different traits. So our intuition or goal in this project is to do GWAS on this index or / quote unquote attractiveness, which is obviously impossible because I don't know how to quantify this. I can't measure this in UK Biobank. So this function can be really complicated. So the magic of this study is really that we realize this is actually possible. The intuition is following. If you understand intuition, you understand 80% of the project. So given this index-driven assorted mating model, here's the intuition. If I have a partner who is, say, tall, let's say it's a socially favorable trait, what does that fact say about me? Okay? So the intuition is if I have a tall partner with this specific socially favorable trait, it may mean that I'm tall. It could also mean that I have great personality. It could mean that I'm super jacked. It could mean a lot of things. What it essentially means is I am attractive. Something about me attracted a partner with a specific trait. The intuition here is you can actually work out the math. So if you measure your partner's specific trait, that actually quantifies your overall attractiveness. And that choice of trait actually doesn't matter. As long as it's one of the traits loading onto the index, this would always work. So this makes this problem empirically testable because instead of running a GWAS on something you cannot measure, all you need to do is to do GWAS on partner's phenotype. This is a third example of GWAS on family members' phenotype. It's a spousal outcome. So we did that in 4 different cohorts. Oh, this is just the math. So essentially, for example, this is wife's height. This is husband's genetics. If you want to do a GWAS on wife's outcome using husband's genetic data, this is actually just a rescaled version of the GWAS on husband's index. You can actually work out math under some assumptions this is true. But then, so based on this idea, we did GWAS on BMI, height, income, EA of partners' phenotype in 4 different cohorts. We collected over 100,000 couples, pairs, so total sample size 200,000. We did meta-analysis. So there is some evidence for this. So for example, BMI and income are very different traits. If you look at the genetic correlation between BMI and income, it's negative 0.27, really moderate. But the genetic correlation between partner BMI and partner income is very high. You can see the number, it's over 90%. This is saying that BMI and income are very different things. Partner BMI and partner income are the same thing. It's about your attractiveness. So, but this is not close to 1 or -1, so we expanded the statistical framework a little bit by allowing some heterogeneity. So instead of purely relying on this index-driven sorting, we also allow trait-specific sorting. If you go to college, most people you know in your life will be college graduates. Even if you don't find them attractive, you are more likely ended up being with someone who's a college graduate, right? So specific traits, such as education, can have a trait-specific correlation between couples that's not going through the index. So we actually allow that. if you work out the math, if you allow trait-specific sorting in addition to index-driven sorting, basically the GWAS on, for example, your partner's education will have a second component that's essentially trait-specific GWAS. So we have to modify the framework a little bit by doing GWAS on 4 different spousal outcomes, but then learn about the shared component across 4 different outcomes. That is actually the index GWAS that we really want to obtain. So we did that. So it's a GSAM application. We did the index latent factor GWAS for 4 spousal outcomes. This is what it looks like. Actually, it has individual level, not individual, but SNP level significant finding. GWAS overall looks okay. Genetic correlation is super interesting. Strongest genetic correlation with education. Some of the physical attractiveness characteristics actually do not show up very substantially. It looks like the education cognition thing that's driving substantially on the index. But then one interesting outlier is actually autism. Autism is a trait that is very well known to be correlated with higher cognitive performance and educational attainment. But given the fact that education is so strongly correlated, you expect autism to be also strongly correlated. It's not at all. It's very null. So this is saying that autism may be correlated with higher school performance, but it does not increase your value for this index. So, but this is a kind of figure you can stare at for hours. There are many results based on this. In addition to that, we did sex stratified analysis. We did country stratified analysis because we had sufficient samples from UK Biobank, but also mobile, which is in Norway. So then I wouldn't get into all the details, but then for specific phenotypes, you see some heterogeneity. For example, essentially this is saying that BMI seem to load more onto female index value, less so for males. Men are shallow, especially in Norway, actually. If you compare Norway with UK, this is big difference. But if you learn about this shared component, which we call the index, it's surprisingly robust. Between male and female, the genetic correlation of the index GWAS is essentially 1. Between UK and Norway, the genetic correlation is 1. So it's a surprisingly robust genetic component when you do the GSAM exercise and learn about the shared component across various specific phenotypes. So this is even surprised us, but it's good to have a robust finding. Final thing I want to talk about is that now that you have this GWAS on this latent trait that drives partner choice decisions, right? So you can do GWAS by subtraction to take out this component from every GWAS out there, for example, education, and then reevaluate the genetic correlation out there based on uncorrected GWAS. So if you do that, you realize a lot of substantial genetic correlation will be driven back down to 0. One example is height and education has been robustly estimated to have a positive genetic correlation, which doesn't really make biological sense. Height is genetically correlated with better school performance. This is, a lot of papers have interpreted this as assorted mating. Indeed, if you do GWAS by subtraction, take out the component that's not driven by partner choice, then the genetic correlation goes back to 0. But another example is if you look at genetic correlation between education and cognitive performance, if you do this, the residual remains highly significantly genetic correlated. That really doesn't change much. So some traits, genetic correlation remain as is, but then some traits, genetic correlation are substantially attenuated when you do this. If you do this for sibling GWAS-based genetic correlation, which is supposed to guard against assorted mating by study design, you do not see this attenuation. Although sibling GWAS in general is very noisy, after doing this, you get very consistent genetic correlation estimates afterwards, which seem to show that this approach is working. So summary, first I have a GWAS on this latent index for partner choice, really enabled by interdisciplinary collaboration of statistical genetics people and economists, and found this surprisingly a consistent genetic component between sexes and between at least 2 different countries in the future. Of course, we're interested in expanding that to even more environmental context. And we even provide some solutions to potentially reduce assorted mating-driven biases in complex trait genetic studies. And this is the 3rd paper in this series of GWAS of family members phenotype. I really think GWAS of family members phenotype is an incredibly fun study design that can give you a lot of interesting results. We have a paper, like I said in the beginning, this is driven by Qingwen, who was an undergraduate student who will also move on to do biostatistics PhD at UNC Chapel Hill. And this is also made possible by Sjoerd from Netherlands, Tokyo from Norway, and Eduardo from, he's from Italy, but he works in Belgium, and a lot of people. I'll stop here. Thank you very much.
Okay, we have a few minutes.
Okay, it's a very interesting topic, and personally, I find this very appealing. So my question is, like, as economists, we actually in the marriage market, we will differentiate the horizontal sorting versus like the vertical sorting, meaning that in this case, for example, the husband may looking for the BMI on website more, and the wife may looking for the income more, like in the marriage market, right? So one problem, I mean, not exactly the problem, but one thing to think about, you index every GWAS. Somehow, I'm not sure how to think about this gender asymmetry in terms of the
Great question.
yeah, attractiveness there.
I'm not assuming that the function for men and women are the same.
Okay.
Right? To increase statistical power, you want to combine everybody.
I see.
If you feel comfortable with the assumption that this is the same function, right? But we did sex stratified analysis, essentially, which is to just regress wife's outcome on husband's genetics and stop there. Do not combine it with the other, version of this. Then you can actually see how different the GWAS look between, male index and female index. Part of my point was, surprisingly, this gives you a perfect genetic correlation. So this is empirical evidence that there doesn't seem to be a lot of difference when you do this GSAM thing to learn about the shared component across multiple spousal outcomes. But for individual phenotypes, you see a lot of differences.
I see what you mean, yeah. Okay, thank you.
That's a great talk. I was just curious if your analytic framework was flexible enough to incorporate same-sex couples in this analysis.
So there's nothing about the statistical framework that would bar that from happening. It's really pragmatic issues about how do you find couples in these biobanks. For example, in UK biobanks, they actually do not provide spousal status at all. You have to impute that. One of the features you have to use to impute this is different sex couple. And basically, different sex, look at the age, live in the same place, same number of kids, same number of cars, your number of variables. So this is an algorithm we rely on to find couples, for example, UK biobank. In some other biobanks, it is totally possible because you can actually directly link people in terms of their relationship status. So in the future, that will be a very interesting direction to explore. Right now, we're a little underpowered for that.
I love this. This is exactly the kind of work that scratches an itch that I have. So thank you so much for presenting this. There was a lot to take in, and you were kind of speeding from it, and I'm going to have to read the preprint to really pour over this. But I'm curious, and maybe you flew by it, but what are kind of the biological underpinnings of this index trait? That's a big conglomerate of a lot of different things. That wasn't clear to me. Because in particular, when you said, I don't think there's any biological reason why education and height would be correlated, but in fact there are biological reasons. So and the fact that it kind of came out in the wash was interesting to me.
So biological underpinning of this is essentially that individual characteristics have genetic basis, and there's a function to link all these 150 phenotypes to the index. That's why this one becomes heritable.
I guess maybe I should have been more specific. So one of the downstream analyses you can do for a GWAS is to kind of figure out the genes that are kind of showing signal in this gene, which GWAS, where are they expressed? What tissues are they expressed in? So
strong heritability enrichment in central nervous system, which is not surprising given the strong genetic correlation with education. So this is, again, a different version of essentially what they're saying, trying to interpret what is this GWAS by comparing the genetic association across million SNPs with other GWAS, right? But the strongest correlation is education, I think it's 85%, something like that. So this looks like education, but not exactly education. But a lot of other things also pop up.
Thank you.
Yeah.
We have one more minute if someone has a quick question. No? Okay, so thank you
very much.
Thank you for the speaker. Okay, guys, let's start our next session, please. Take your seats. Thank you. Thank you. So the next section is another method session with Dylan from UCLA, Weilong from Cambridge, and Gay Yu from Purdue. So Dylan, please start us off.
All right. Hi, everyone. I'm Dylan Steinica. I'm a PhD student at UCLA, and I'm working with Daniel Benjamin and Patrick Turley, and also on this project is Jonathan Jalla. And we're going to talk about our project using siblings for more precise and accurate heritability estimation. And the two goals here are to figure out how to switch slides. Do I have to point it somewhere? Oh shoot, where was I pointing when it worked? Okay, well, it worked on this slide. So the first goal is developing a heritability estimators that are more precise and accurate. And the second one is going to be clarifying some ambiguities that we believe are underappreciated when you're applying heritability estimations for families, and something to be aware of when you're trying to estimate heritability and you're comparing families or non-families, and to see how to interpret that. Where am I? Okay, it's pointing here. Okay, I'll try that. Okay, yeah. I won't shoot the laser pointer at anyone though. So first, before we get into the estimators, background on heritability, let's think about a phenotype as being driven by genetic effects. So that's a person's own genetics, the effect on themselves. And then it can be also parental genetic effects. So that's a parent's genetics affecting the parent, which affects the parent's phenotype, which the parent's phenotype could affect the child. So that's an indirect genetic effect. And then there are other things we'll just throw into the residual, and that'll be things like environmental noise and other things. It could be like the grandparents' genetic effects. Oh, okay. So heritability, what we care about, though, is the part of the person's own genetic effects, so the variation in the genetic effects over the variation in the phenotype. And if all the variation in the phenotype is explained by variation in genetic effects, heritability is going to be 1. If none of it is explained by variation in genetic effects, heritability is 0, and it ranges in between. And typically, this is focused on the population level. When you look at all genetic effects, is the broad sense heritability. And if you look at just the additive genetic effects, so that which can be explained by a linear model, that'll be the narrow sense heritability. And for complex traits, the narrow sense heritability is expected to be very similar to the broad sense heritability, and it's easier to estimate. And the other heritability is going to be SNP heritability. This is the additive genetic effects, which are tagged by certain genotype SNPs that you have. We're going to be focusing on these two latter heritabilities in the talk. When you want to estimate heritability, though, you want to avoid bias. Some of the most common biases due to population stratification, indirect genetic effects. These are just different instances of gene-environment correlation, because you don't want the effect of the environment that correlates with the genotype. You want to know the effect that is caused by the genotype, and it may be mediated through the environment. But when you use population-wide data, you're going to often get a lot of that population stratification. You can control for that with PCs, but still not perfectly. And even still, you can't fully account for these indirect genetic effects, like from the parents. A way you can control for almost all of that is by using siblings. But if you use siblings, you have a lot less data, so your results are often less precise. There's two trade-offs here to try to work with. And to explain how siblings can how siblings can reduce some of the effects of bias here, I'll explain this. So we have the same formula for the phenotype. I'm going to switch it to math. Same exact thing, even though it's written in LaTeX, so don't get too scared. And the first part is X is the sibling genotype, and then the beta is the effect size of that genotype. And the other part is the parent's genetics, and then the environmental noise. If you just regress this genotype onto the phenotype, or phenotype onto the genotype, you won't estimate that direct genetic effect, probably. It'll pick up some of those other effects, so that population stratification and some of that parental genetic effects. But if you take the difference between two siblings here, the parental genetic effects will cancel out, and also the difference between siblings is not going to be due to ancestry, it's just due to random segregation, just due to meiosis. So any variation there, if you estimate the differences, phenotypic difference onto the genotypic difference, it'll just estimate that genetic effects that we're interested in here. if there are no sibling indirect genetic effects. But for the most part, it'll remove that bias. If you didn't follow any of that, all you need to know is that sibling differences make estimates less biased. And we're going to be talking about 4 methods here that take advantage of the sibling difference to remove that effects of the bias. And they're all going to, or the first 3 are going to be in the form of siblings phenotypic similarity on the left-hand side and the siblings genotypic similarity on the right-hand side. And it's in a regression to estimate the heritability. The first one is a method that's been around since 2006. It's Vischer's so-called sibling regression. It's based on IBD. And on the left-hand side, you just have the difference between 2 siblings' phenotypes, and you square it. And on the right-hand side, you have the siblings' IBD genotypic similarity. And this regression estimates the narrow sense heritability. A similar method that we're introducing is we're calling within family sibling regression. The difference here is that instead of IBD on the right-hand side, using IBS. So the difference between siblings on their genotypes is on the right-hand side for IBS. And then the next method we're introducing is not just looking within a family, but it's going to look between families as well. So it takes 2 siblings' phenotypes, takes a difference between them, takes 2 siblings in another family, takes a difference between those, and multiplies them together. And then that will help you estimate the heritability. On the right-hand side, of a similar concept, but it's with the genotypes. And by doing this, you compare across every possible pair of families, so your estimates can be a lot more precise. You're exploiting a lot more of the variation across the population. And then it'll be estimating SNP heritability, it's just the way it works due to the LD across the population. And we'll be going into the differences in all these methods through the rest of the talk. And we're also going to be comparing it with sibling LDSC, so use a sibling GWAS on LDSC, and this is in the same category of these between family methods. So we have the first 2 methods with our within family, and then the second ones are between families. Here's our first results, first results here on a simulation, and we see that the IBD-based sibling regression and the IBS-based within family sibling regression are very similar in their precision and how accurate they are. And the next part, you see the 2 on the right-hand side, your right-hand side as well, is the between family method, so between family sibling regression and LDSC. And between family sibling regression, both of them are a lot more precise, but between family sibling regression is accurate, it's not biased here, whereas the LDSC is biased. And the reason we believe is because of the relatively low sample size. That LDSC has a finite sample size bias here. And even though we're using all European ancestry siblings in the UK Biobank, it's still not enough for LDSC to produce confident results. So the between family method's more precise and more accurate here. And we'll be going into some of the differences further. It's showing the real world results of real phenotypes in the UK Biobank, and using 3.5 million SNPs. The first 2, the orange and the green estimates are the green one is the IBD-based sibling regression, and the orange is the IBS-based sibling regression. And those 2 methods perform very similarly across these real phenotypes as well. I won't be going into too much about why you care, who cares about the IBS one here. There's some other reasons why we might, but for the scope of the talk, we'll be focusing elsewhere. And the between family method you see is a lot more precise. The standard errors are much smaller, often about like 4 times smaller. But another thing to note is that the estimates are a little bit smaller, and this is because it's estimating SNP heritability and not narrow sense heritability. So the 2 things that we're going to be focusing on, why these methods differ and why all within family versus between family methods are going to differ, is due to LD. So the effects of LD within a family will allow you to tag narrow sense heritability, while the effects of LD across families or across the whole population is going to cause your method to tag the SNP heritability. That's because the LD within a family is a lot stronger. So if you have 2 siblings have the same genotype SNP, there's a good chance they share a huge chunk of the chromosome nearby together. But if 2 random people in the population have the same SNP, maybe they'll share a few SNPs nearby, but often they won't share many, so that will tag just a few of the SNPs. Here's a demonstration of that, showing the between family method on BMI. As you slowly add more SNPs, you see the heritability estimate increases. It's tagging more of that variation. But for the within family method, once you just add a few SNPs, that jumps up to tagging almost all the heritability. It's because it tags those larger chunks of the chromosome that siblings share together. As more SNPs are genotyped, these estimates converge somewhat, but differences may persist if there's non-genotype rare variation within a family, and also due to gene-environment interactions that may differ within a family versus across a population. And that differences in the gene-environment interactions would also add another dimension that separates these classes of estimators. So heritability is typically focused on average across the populations, so there's no like common gene-environment interactions that are being picked up here within a family. But if you're just comparing within a family, then you might have an additional common environment that's mediating genetic effects more similarly for siblings than it is for people across a population. And to explain that, illustrate an example. Let's say we have these 3 genotypes which happen to correspond to different colors of green skin. These Martians just invaded Earth, and they're settling here, and they have a different susceptibility to skin cancer. And some of them settle up where there's low UV environment, and the first 2, they have never have skin cancer, and the last genotype always gets skin cancer up here. So all phenotypic differences are explained by differences in genetic effects, so the heritability is 1. Another family of Martians settles down the high UV environment. The only difference here is that the middle green person, now that genotype will always get skin cancer. Their genetics isn't protective down here anymore. They're the same variant, but in a different environment has a different effect. So here still the environmental or the phenotypic variation is completely explained by genotypic variation, genetic variation, genetic effects. So the heritability is also 1. So this, if you look at within family estimates, you'll get a heritability of 1 here. This would include if you do something like a twin study among them. However, if you just change your perspective here without changing any of the biology, or you don't touch any of the experiment, you combine the populations. Now the middle green person, half the time this genotype corresponds to getting skin cancer, half the time it doesn't. So all the variation in the phenotypes here isn't perfectly explained by variation in the genotype. So some of it's explained by the environmental variation. So now the heritability is going to be less than 1. Nothing changed. It's not that this estimate's biased, 1 isn't. It's that how you frame the question, and are you accounting for that common environment, or are you averaging out all those environments across the population? Another way of looking at it is in that equation there, the genetic effects is the genotypes and the effect sizes, and those gene-environment interactions will change those effect sizes. Is the average effect size across the population, or is the average within a family? And just a graph illustrating that last point as well. We simulated different families where all families have the same genetic effect sizes, so you could think they're all on the same latitude for that example. And then that's going to be on the far end of the graph. And then closer to at 0, the family environments are not correlated, so they'll be very different from each other, and they might have very different gene-environment interactions. When we do that, the within family estimates stay the same at the top in orange, and those ones don't change because it doesn't matter what other families are like. All the within family estimates matter is what your family is like and the variation within it. But between family estimates, these will change depending on what the other family's environments or gene-environment interactions are like. And you see it decreases as the environments become less correlated with each other. And it's identical with the within family method if the environments, gene-environment interactions are the same within each family. So the final way of framing it is you see there's 2 ways, 2 things to be concerned about potentially when you're applying your own within family or between family estimates, when you're implementing in a study, or if you're arguing with it on X Twitter about people about which estimator is the best and if it's biased or not, knowing that this might, between families, it might be it's tagging the SNP heritability. Within families, it's tagging narrow sense heritability. But within family methods, many of them will tag that additional common environment effect if it's there. So it may not be there, it may be there, but something to keep in mind. And then twin studies would go along in the bottom here, and then I believe there's other estimates can be categorized in here as well. So just want to thank everyone who's collaborated on the project, and the rest of our group, SSGAC, and all of you for attending and listening. Thank you.
Questions?
I could ask one question. So, I'm kind of very interested in the heritability in the GBI environment. And for the example you gave, if you think about these two type of household, and we are thinking about one fatal disease, right? So you can expect like the family size could be quite different. Like the one with this particular SNP that has not protected by the healthy one, they may just have smaller family size. And when you mix them together, then there will be like combination differences just due to like the heritability in these two different situations, right? So should I worry about that?
Potentially, it depends. If you, I mean, most of the, in the UK Biobank, I think most people don't have a sibling, and then if you do have a sibling, you should have one of them in there. So I don't know how much variation to say in here there even would be in family size. You get to genotype, and it depends, is it like a childhood onset disease, or does it affect the family size? So I think, yeah, it would depend on the phenotype. It's something to keep in mind. And yeah, we were thinking about, so I guess it falls in the category of every participation bias thing as well, but important, especially if I'm saying the Martians have 100% like incidence of skin cancer on certain genotypes. So thank you for noting that.
Other questions? Okay. Thank you, Dylan. Let's move on to Wei Long.
Okay, thank you so much for including this paper in the program. Honestly, I really appreciate and enjoy the conference so far. And the things I learned from these 2 days actually is more than the things I learned from past year. So the only thing I feel regretted is like I didn't really join the community earlier, but I will definitely continue to do so in the future. So this paper, we will talk about the genetic endowments and lifetime earnings and understanding mechanisms. And it's basically a home team with my colleague, Eric and Varun, and our student Chang Yu, and Uta is coming from University of Bristol. So let me start with give you some motivations. So there will be a lot of evidence about the importance of genes that can predict different lifetime outcomes, right? And a lot of use actually contribute a lot in this literature. So if your name is not on list, it's because like there are too many, and sorry, I don't really including them, but more in the paper. So we see the genes has very good predictive power on skill formation in the childhood period. And then we also see it has a good prediction on education attainments. And then it will also contribute to the labor market outcomes and earnings. But if we really want to think about something about more complex traits like lifetime earnings, then the mechanism, how exactly the genes can link with lifetime earnings become much more complicated, right, in terms of mechanisms. And all these kind of things we listed before will become something very relevant in terms of as a mediator or as a channel enter into understanding this correlation. So this is where we try to contribute, and we want to actually understand the effect of genes on the lifetime earnings from birth to retirement, and try our best to quantify the importance of each channel in this process. So, of course, it's a very aggressive or a bold argument to think about all the possible channels. So we try to take a first step and think about like two basic channels we feel like that are very important. So the first channel we call it skill production channel. So this is like just thinking about the genes will naturally play a role in your skill formation. see through the mature of the brain development, right? So this is a more biological role we will think about genes may play in this skill formation, but actually holding the choice or environment to be more constant. And then the other type of channels we feel like it's also equally interesting or even more interesting. It's like the choice equation channels, we call this. So essentially, you will feel like people will be strategic in terms of their choices. And the genes will play a role to think about how people's behavior may respond to their endowments, right? So when we think about this choice, because we are trying to model people starting from the birth to retirement, so there will be many choices are in play. So when people were young, then maybe their parents are play a very major role in terms of the choices. So here we want to include in the parental investments as well as the school quality investments as like the major choices in this period. But when they become adult, then there will be more sequential choices such as education or occupational choices. You can think about naturally all these choices matters a lot in terms of determining your occupation trajectory, your career trajectory, and therefore contribute to a lifetime earnings. Right, so we actually try to model this as a more sequential structural model. So I don't really list the very tedious equation here, but just using a graphical representation in terms of how we think about this process. So supposedly we have someone who were born with specific genetic endowments. So the two type of choices we listed is using different color of arrows. So the first type of choices, which were the skill formation channels, we list on the left-hand side and using these green arrows to show its channels, right? So we have multiple measures about their skills at different stage of life during the childhood period, and then it will eventually contribute to your final skills and therefore contribute to your earnings. And on your right-hand side, those ones using the blue areas, so arrows, these will be the sequential choices we are interested. So when the individuals were young, then it mainly is parental investment decision that are play the role. And the investment, we also have these repeated measures at age 7, 11, and 16. So, and then there will be a further choices made by individuals about education and further occupations. So one thing I definitely want to highlight is like there will be very interesting interactions between these two type of channels. Like I will just give you two examples as illustration how this model works. So the things we want to actually focus is the potential interactions. So let's say if I want to understand the correlation between the initial genetic endowments and lifetime earnings via the channel that the genes may affect the skills at age 7, right? Then there will be like two different type of channels we want to highlight. The first one is like mechanically or biologically, when your skills at age 7 become different due to the change in progeny scores, this will also shift your further at the later skill levels, and that one will have a direct or like a more primary impact on the earnings. So this we are using the solid arrows to show the mechanism. And more importantly, if someone's skills become different, right, the parents will have this kind of so-called behavior responses. So may think I want to reinforce my investment because my kid seems more promising. So I expect that they will bring better earnings in the future. So my investment gives me higher returns. So that's why they may induce the investments from their parents. Okay, so this is one type of interactions. And on the other hand, if you think about interactions coming from the choices, they will also have some kind of differentiation in terms of the primary effect and then the indirect effect. So let's take a look at the progeny scores on the affecting the earnings via the channel of education, right? So one thing that is more directly is that with someone who has higher progeny score, especially for education attainments, there will be a very strong predictive power in terms of education levels, education attainments in the end. But such kind of higher education attainments may have two possible channels affecting your earnings. One is more directly, so because people are productive, that's why they become higher earners. And the other one is relatively indirectly. It means like when you have higher level of education, you may choose like those more lucrative occupations, and that also further contribute to your earnings, right? So we want to differentiate those kind of channels as well. Right, So the reason we can do such kind of decomposition actually also rely on the data availability, right? So we are using this NCDS data, and which was individuals born in 1 week in Britain in 1958. So just comparing with very commonly used biobank data. So our advantage is that this is a panel structure. We are following them from the birth to retirement, so we will have their realized lifetime earnings. So we are not really guessing their earnings individually. We actually can calculate what is their earnings over their lifetime. But the disadvantage, to some extent, is like we will have much smaller sample size. So for example, we are not able to exactly apply in the within sibling GWAS, and we have to apply the population GWAS. So that's kind of the limitations of the sample size. But be that as it may, so we will have a bunch of measures during the childhood period, and we have a bunch of measures during the adult period. So that's why we can having those kind of channels in the model to quantify their relevant importance. Okay, so there's just one graph as a summary statistics to show you why we feel this kind of channels would be relevant. So the way we are doing this is actually we distinguish individuals by their Portgen scores, and we chop them by half. So we are comparing with the one with high PGS versus the one with low PGS. So the individuals with high EAPGS, actually they are more likely to start with a better family environment. So their parents are more likely to have higher SES. And then in the childhood period, they actually will receive more time investments. So this will be the parental investment directly in time, right? But they will also having, the parents will also send them to the school with better quality. And then we also find that they will have a better cognitive skills, and we also have non-cognitive skills, but that is a little bit less significant. And then in terms of the education attainments, we see like they also have more years of education, and they are more likely to work in a more lucrative occupations and working more hours. So all these things actually can see are kind of contribute to the higher earnings. Thank you. So that's exactly the reason why we want to having all these kind of channels in the model. Okay, so let me show you the first result we find. So in terms of the total effect, we find like 1 standard deviation increases in the EAPGS actually can contribute to the increase of lifetime earnings for 19%. So this is actually a quite non-trivial number. But in terms of the channel decomposition, within this 19%, we find that about 10% comes from the choices channels, like your sequential choices over life cycle, we demonstrated. And the education essentially becomes the most important choices among all choices, but which is kind of not surprising because we are using the EAPGS. But on the other hand, we see like the skill formation channel also contribute to this 8% or to 9%, right? But one thing I want to actually highlight is when you think about this 9%, it's not exactly that mechanical or biological because we also account for the interactions. So meaning that whenever you have higher initial skills, you may also induce more parental investments. So there will be some kind of choice component also enter into this skill formation channel. So that's why we feel like even with the genes seems like more predeterministic, but we see like there's a lot of contribution coming from people's sequential choices that build up the link between genes and lifetime earnings. So that's definitely something we want to highlight. And then in comparison, for this family genetic association, this account about like 4% of the earnings. It's just like a reference group we want to compare. Okay, and the other thing we also kind of very interesting is trying to understand the heterogeneities in returns to investments. So we kind of try to leverage the advantage to having this dynamic model, meaning that we will have a sequential investments in terms of human capital investments at different stage of life. Okay, and what we are doing here is we re-estimate the model, separating them by the individual with low PGS and individual with high PGS, and estimate these coefficients. And now what we want to highlight is that there's kind of seems like a changing in terms of complementarity between the EAPGS and then these kind of investments. And more specifically, during the childhood period, we see that the parental time substitutes for low genetic endowments, right? So there's a substitution in the childhood period. So the one with low PGS actually get higher margin returns in those kind of investments. But if we are looking at the later education attainments, showing blue here, then we see this kind of pattern reversed. The education attainments actually complements the higher genetic endowments. So the one with higher advantage in terms of their genetic endowments actually also get higher margin returns. So there will be a very interesting policy implication, because if you really want to mitigate the initial genetic differences, it seems like we identify the most vital period is during the childhood period, because that is a period you have this substitution rather than complementarity. Okay, so the other thing, or the last thing we report is actually try to understand what could be the potential upper limit that the genetic variation can contribute to the lifetime earnings. Because we only start with the EAPGS, but we also acknowledge like this is only a small portion or a specific portion of the genetic variation, right? So in order to do so, what we are doing here is kind of following the GWAS by subtraction method, but we sequentially following the, using 54 different GWAS and try to see if we adding more GWAS or adding more polygenic scores, is there any kind of additional variation that can pick up by this new GWAS that was not in the EAPGS? Okay, so the left-hand side will be those kind of other polygenic score, their marginal contribution to the earnings in terms of their marginal effect. Okay, and then the right-hand side here will be their contribution in terms of incremental R square. So interestingly, although we have this relatively large set of candidates, the education attainments actually play a very important major role in terms of explaining lifetime earnings. But the other relevant GWAS or polygenic score, including the mental health, agreeableness, restorance, and also household income. So some of them will have much less intuitive channels than the education attainments, but we definitely want to show them for completeness. Okay, so let me just conclude. So this is actually an effort we're trying to think about the contribution think about the association between the genes and lifetime earnings. And in order to do so, we develop a life cycle model. I only show you the graphical representation, but if you're interested, you can check the paper. And then we find that there will be two major channels playing a role. So one is through the sequential choices equations, and the other one is through the skill formations. And in terms of the explanatory power, then we find like about 4% of the lifetime earning variation can be attributed to the EAPGS. And if we are adding possible EAPGS we can find in the literature, then it will kind of increase slightly in terms of total variation that can be explained. But and lastly, there's a policy implication, because if we really want to, mitigate the initial genetic differences, it seems like an early child investment will be very important. And because this is like a crucial period, we see the substitutes between the genetic endowments and the human capital investments. That's it. Yeah, thank you.
Thank you. Thank you. Thank you, Wei Long. This is very interesting. I'm going to use my privileges as the chair and ask the first question. So for me, the dichotomy between the skill formation and choices is a funny one, because you'd expect the skills to drive choices, right? If you're more skilled, your cost of getting more education is lower, right? And it seems to me the way tell me if that's right, the way you are separating those is just some assumption on the timing of this. So that's kind of something that, what is the assumption that allows you to separate these two? Because for me, they're very interconnected, and I'm not sure how we can actually measure those separately. The other comment I had was I really would advise you against talking about skills as the biological channel, because we know the EA, has a lot of other channels. Even if you do like direct genetic effects, there's so many social channels. So kind of calling that biology is something that I think we should be very careful about doing. But super interesting thing. So yeah.
Yeah, thank you for both your comments. First of all, I completely accept your second comment. So for biological channel, we only try to emphasize like there's a biological component, but we fully understand like it could not be biological that much, right? So back to your first point, The reason we can distinguish, we can specify, the diagram in this way, essentially, of course, there's a timing assumption. So we will only, there will be a value function, value added equation there, and there will be a AR process there. But of course, we have to make a decision whether your current investment will only affect your future skill levels rather than contemporaneous one. So that was the assumption we were making. Yeah.
Okay.
Great presentation. Just to follow up a bit with that. So I think throughout your presentation, maybe I'm wrong here. I think you are making assumption that the skills and the parental investments are kind of positively correlated. So basically, parents making investment to promote children's skills. But I think in reality, as a parent, I know it could be the opposite, right? Because the parent may making investment due to children's deficiency or lack of skills. So I wonder if you take that into account in the analysis, or
that's a very important point. Actually, we don't really pre-assume the sign, like how the genes will contribute to the parental investments. So it could be the reinforced channels we've emphasized. Of course, we also allow the possibility that maybe the parents want to play the fair game, so they always help the children who left behind. So the estimates will tell us it's a very positive, significant sign number. And we also check literature, like Hallmark et al. They also do something very similar in the childhood period, and their estimates are also positive and comparable. So in general, I think we find some consistent evidence, like there's the reinforcement effect rather than play a fair game.
Very interesting talk. So I'm just curious about the data because I didn't know about this data set. So it's nice that you have several time points. So can you talk about any potential loss of follow-up, and what are potential factors? Would there be any bias associated with that?
Okay, thank you. Yes, this is actually a very important point. Thank you for the question. So definitely we will have a lot of attrition concern, especially we try to follow the individual for such a long horizon. So in that case, like many people may miss in the middle over these 60 years. So one thing we definitely checked is about the balance in terms of, okay, so this attrition to some extent is something we are inevitable. But the other thing we may even worry more is about how to get this genetic data, because it was collected in the middle age of their life, like about in the wave of 40 when the technology become available there. So we only have a subset of individuals that will consent and we can find to having this genotype data. But what we did is a comparison between this subsample who get with genotyped versus a full sample, and there's some kind of difference in terms. For example, the genotype sample has more education, and they also have slightly higher earnings. So there will be like a little bit like representation issue, representativeness issue. So external validity, we acknowledge the limitation there. Yeah.
Thank you, Wei Long. So next we have Geyu from Purdue. Thank you.
Good morning, everyone. So my name is Geyu Zhou. I'm from assistant professor of Department of Biological Sciences from Purdue. I'm also part of the Purdue Social Genomics Initiative. And thank you for including my paper in this program, and thank you for organizing this wonderful conference. Today I'm going to talk about the polygenic prediction in the admixed population, and this work was primarily done when I was a postdoc at the Yale with Hongyu. So we know that the polygenic risk score or polygenic index, PGI, they are basically the predictions of the complex traits based on the genetic variants. And there's one emerging issue of the PGS or PGI is the transferability across the populations. So for example, if we take the prediction accuracy of a PGS or PGI in the European as a reference level, and we see that the accuracy really decrease when we are focusing on other populations, for example, the East Asian or Africans. And this issue is particularly worse for the admixed populations, for example, for the African Americans. And in the past 5 years, there have been ongoing efforts from both the data collection and method development side to try to solve this issue. So from data collection, we have Paige, MVP, and all of us. They are collecting more and more admixed individuals into the GWAS study, but also creates opportunity to develop new methods to analyze the data. And traditionally, we know that people trying to view the population as a discrete genetic clusters, but there's recently paper advocating that we should move to another direction to rather viewing their populations as concepts from the continuum of the genetic ancestry. And if we look at, again, the polygenic prediction accuracy is kind of the decrease if it's trained on a homogeneous groups and transferred along this continuous ancestry. Now we talk about the admixed populations. So admixed populations, it arises from, so for example, one of the parents is from European, and another is from the African. And the first generation will inherit one chromosome from the one parent. So one chromosome from European, another from the African. But after many, many generations, because of the meiosis during the recombination during the meiosis, the part of the genome segments, they will originate from the European ancestry and the other parts are from the African ancestries. And we typically refer to this as a local ancestry as comparing the global ancestry components. And the left figure, this is a PCA of the genotypes from the two largest admixed groups in the US, the African American and Hispanic Latinos. And you can see that they are from the PCA, they're really continuous variations along the axis. It's not like European or African. And this brings up how we can best construct PRS for those admixed individuals. So basically, in the past few years, there have been two strategies. The first is to really utilize a large sample GWAS summary statistics and the linkage disequilibrium reference panel from two populations. For example, one can be the European, and another can be the East Asian or African. So you can jointly analyze the data from the two populations using these kind of methods. But these methods make different assumptions, but they all use these two types of data sources to fit a model. So these methods are mostly designed based on the assumption that you can divide the populations into a more homogeneous or discrete groups. So this might be a problem because, for example, the LD, we know the patterns in the admixed populations can be a little bit different with homogeneous populations. And the second is the sample size of the GWAS from the admixed populations are still quite limited. So this brings about our more traditional approach by analyzing those individual level data, assuming that you can fit a model based on the phenotype and the genotype alone. So this includes the traditional GWAS methods like BSLMM and more recently developed methods to fit in the admixed individuals like IPGS and Gaudi. And these methods, they work directly with individual level data to account for the continuum of the genetic ancestry. And one problem is, can we use those local ancestry to help us to design those PRS methods? Or is that helpful to improve the prediction accuracy? And here, I'm showing two kind of the scenarios or assumptions can be made for this local ancestry. The first is the effect sizes, they are somehow different across the local ancestry. So think about this first allele, they are both A in the European and the African, but their effects, they are different. And the second scenario is you don't need local ancestry, you can assume that the effects, they are same across the ancestry. So under the different ancestral effect size, we can describe the linear relationship of the phenotype regarding their ancestral dissolved genotypes, also along with the covariates like age, sex, and PCs. And we are mostly interested in estimating the ancestral effects across the local ancestry. And more traditional GWAS, you can assuming that those across ancestry effects, they are same. So like you only need to estimate one effect to derive the polygenetic scores. And in our work, we propose a method called SDPR admix to really take these local ancestry into account. So this is the first part of our model or the equation. But we go beyond that to assume that the effect size of the across local ancestry can be divided into 4 components. The first is the effects. They are really both 0 across the 2 ancestry. And the second component, these effects can be ancestry 1 specific, which means their effects are 0 in the ancestry 2, but non-0 in ancestry 1. And similarly, for the third component, the effects, they are ancestry 2 enriched or specific, which means their effects, they are 0 in ancestry 1, but non-0 in ancestry 2. And the last component, they are somehow correlated across ancestry. And we model the second, third, fourth component using a mixture of the derivative process. So you can think of it's a mixture of normal distribution. The advantage is this is non-parametric. Yeah, you don't need to do some more parameter tuning, and it's really adaptive to accommodate both large and small effects. And after that, we can fit the model and output the effects to derive the score. And we compare our methods with a couple of other methods. The BSLM is a traditional GWAS methods, which does not assume the heterogeneity of the effect sizes. And it also does not using local ancestry information and use all of the genome-wide SNPs. Gaudi and IPGS, they are more recently published and more tailored for the adnexed populations. So Gaudi also assumed there's kind of effects across local ancestry, but it's used fused lasso to do both variable selection and encourage those shared effects. But the drawbacks of Gaudi is because of computational issue, it has to do the P plus T, the pruning and shielding first, and it's not using all of the genome-wide SNPs. IPGS is more efficient implementation of the elastic algorithm, and what they do is to also include some heterogeneous effects directly if they find such effects is present in the data sets as a covariates. Partial PRS is a very naive approach, which basically deconvolutes the genotypes into 2 ancestry and using the effects from the GWAS summer stats to derive those scores. It also just using P plus T, so not accounting for all the genome-wide SNPs. And we basically first train our model using the 2 data sets. The first is PAGE. That's when we developed methods, and when All of Us version 7 came out, we also switched to All of Us. And based on these training data sets, the average global proportions of the European, African, and mixture is like 80% of the African and 20% of the European. And for the testing data set, we're using UCO Biobank to select those individuals, again, with 80% of the African and 20% of the European. And this is the 4 traits we have in common in all these 3 data sets. And we can see that first, the sample size of All of Us, they are about much bigger than the PAGE, but the SNPs, because we're using the intersections of the whole genome sequencing with HiMaps3 SNPs, actually a little bit lower than the PAGE. And this is the first results when we train on PAGE and test on UCO Bank. We basically report the Pearson correlation between the predicted PRS with the real observed traits. So the red one, they are our methods with the correlation estimated by another software, AdmixKit. First, we can see that overall the performance of our methods is better, but we also change our correlation row coefficient to 0.999, so to encourage more strong sharing, and actually our performance is down a little bit. And we also test our performance by training on all of us and testing on the UK Biobank. We can only run 2 other methods here because there are some computational issues for BSLM and Gaudi when deploying on all of us. And for across these SCAN4 traits, really our methods perform better for the height, platelets, and the white blood cell, and other IPGs have performed better for BMI, but overall there's about 10% improvement over IPGs and about 400% improvement over PPRS. Also, we can see that overall the absolute scale of the performance of each method, they are better in all of us compared with the page. And we also look at what our model identified. We can see that across these 4 traits, the effect sizes estimated by our methods, they are really different. For white blood cell, there's more strong kind of ancestral effects, while for high BMI or platelets, there are some of the shared effects across those ancestry. And next, I'm going to briefly talk about how we can extend such two-way admixture model to the Hispanic Latinos. We know that Hispanic Latinos typically have three-way admixture of the European, Native American, and African ancestries. But past studies have shown that the 2 of these ancestry are often predominant within certain ethnic groups. For example, the Mexico mostly are Native American and European admixture, and the Caribbean, they're mostly the European and African admixture. So we think that just using a two-way admixture model may be sufficient. And especially in all of us, most of those Hispanic Latinos, they are actually two-way admixtures of the European and the Native American. But in UK Biobank, there's like three-way admixture, so we just merge the African tracks into the AMR track and apply the two-way admixture model we trained on the all of us to the UK Biobank. And there's a sample size and number of SNPs we used in such model. And we can see that overall, there's a 22% improvement over the IPAGS and 100% improvement of PPRS when we test on the AMR individuals in UK Biobank. So in summary, we developed methods to work with the individual level data of the admixed individuals to construct the PRS with about 10 to 20% of the improvement. And our methods really demonstrate the benefits to include the local ancestry informations and cross-ancestry genetic architecture to train PRS. And our method is publicly available on the GitHub. And I would like to thank my advisor, Hongyu, and my co-authors, Israel and Yuhan, and all the data sets are available for us to train our methods. And thank you.
Thank you. Thank you. Questions?
Thanks. Great talk. So I think you kind of hinted at this, and so maybe I just missed it. But so in your model, you're assuming that there are SNPs that affect one kind of ancestral population and not the other. You estimate these pi parameters. And I was wondering kind of what are them when you fit this model, how big are your like pi 1 and pi 2? Like what fraction are finding? Because it's kind of a weird case that you'd think that like there would be a SNP that like only affects African ancestral alleles, right?
Yeah. So those are actually, I would say, also the ancestry-specific SNPs. So for example, some SNPs, they have the allele frequency higher in ancestry, but higher in African, let's say, and almost zero in European. So that will be assigned to that group. And it's traditionally just removed. If you just look at the minor allele frequency cutoff, like doing a filtering, that will be removed. But our model, we kind of include them as this, the second component. I would say overall, you can see that most of those kind of ancestral specific effects, they actually have allele frequency higher, let's say, in one ancestry. And overall, I would say if the both allele frequency, they are, let's say, greater than 0.05, they're more likely to have those shared effects.
Thank you. In practice, you almost always also have an external GWAS summary stats, at least for European ancestry. So if the goal is just to maximize R-square in a specific admixed population, how would you recommend the approach?
Yeah, so first, in the IPGS paper, they compare their performance with PRSEXS to use the European GWAS with those small sample size African GWAS. And I think they find that for some traits like WBC, the performance is actually better to directly working on directly working with the individual data from the admixed populations. But I have current ongoing work is to add another layer. For example, you can think about there's additional data sets with the European GWAS summary stats. And you can think of it as beta 3 here to incorporate another like sharing information in the model so that you can borrow those large summary stats from European together with the local ancestry dissolved genotypes from the admixed population to jointly fit another model.
Any more questions? No? Oh, okay.
What it is by the time you get to me. I don't remember. I'll ask you later if I think of what it is.
Okay. Oh my God.
Yeah, so would you comment on an alternative approach that use tools like Tractor to do ancestry-specific GWAS first, obtain... By the way, you can also meta-analyze that with external GWAS, and then you have multiple summary stats and work with that.
Yeah, we can definitely do this. But again, I think there's currently those methods to analyze this because if you do a Tractor, if you derive the model, there's kind of, unlike the traditional homogeneous population, you kind of have a cross product of the two ancestry components there. So you need to change the framework. It's not like you can completely apply some methods derived, for example, PRCSX on these kind of summary stats. If there's local ancestry interactions there, you need somehow take them into account.
Okay, so we have 1 more minute. If anyone has remembered their question. Okay. Or anyone has another question? Okay.
It's just about scalability. So you have this with two ancestries, but you could imagine applying it to a sample that also has like a Hispanic population, or even if you have admixed, like African populations, you'd maybe worry that if you have East African and West African admixture within your sample, that you should maybe treat those as separate ancestries as well. And I wonder to what degree this is just going to blow up, right? Because it's combinatorially would get really large.
Yeah, you can think of this as a trade-off between going down into the more refined granularity with the heterogeneity, because you are refining down the tracks. If you include more ancestry there, then on average, the size of the tract corresponding to the ancestry will also be smaller, which may be a little bit difficult to fit the model. So we believe that my practice is to just infer the ancestry component based on the FST. So we need to have some difference between the ancestry in order to such model to work. And I would say generally 2 or 3 probably is sufficient from my point of view.
Okay, thank you. Let's thank our 3 speakers.
So welcome back, everybody, from lunch. Excellent. It's great to have so many people still here. So Kelly Bakulski is a molecular epigeneticist. Let me try that again. Epidemiologist and environmental health scientist who's an associate professor of epidemiology at University of Michigan. She did a postdoc at Johns Hopkins and a PhD from Michigan. Her expertise is in life course, I'm saying her expertise is partly in life course toxicant exposures and how they affect our cells and our health. And in particular, one aspect that's so interesting is how biomarkers collected now might be able to tell us full histories of exposures that are really important for our health. And I hope that'll be one element that you see in the slide here that's going to show us a way in the upcoming years of this type of social genomic research of how to add very important elements that are already in the data but that we haven't yet captured fully to understanding human health. So join me in welcoming Kelly for us.
Thank you so much for those kind words, Jason, and for the invitation to be here with you all today. I'm really enjoyed my experience here at the Advances in Social Genomics Conference. This has been just such an incredible collection of folks with expertise in a diverse array of areas spanning from history to philosophy to sociology to econ. It's helping stretch me in new areas and learn new topics. I've really appreciated this. So hopefully I'll be able to plant some seeds with you to think about ways that your social exposures, these ways that you have a lot of expertise in these factors like education and income, I'll be thinking about a related, maybe downstream factor of chemical exposure. So maybe you'll be able to think about and reflect on your particular social exposure of interest, and I'll show you ways at which that might be related to a chemical environment as well. We are grateful to have received support from the National Institutes of Health and the ALS Association. And I'll focus on tools and approaches that are hopefully applicable across many disorders, but the example one I'll use throughout my talk today is dementia. And dementia is a neurodegenerative disorder that's characterized by your loss of cognition that impairs your ability to carry out activities of daily living. And we consider dementia an umbrella condition of multiple pathologies. So the most prevalent form of dementia is Alzheimer's disease, and some of the additional dementia types include vascular dementia, FTD, and Lewy body dementia. I'll use these terms a little bit specifically depending on the type of study I'm working with. Our clinical-based cohorts often have the refinement of those specific pathologies, like Alzheimer's disease, versus some of our more population-based studies. We don't quite have those diagnostic specificity, and there we'll be working with dementia more broadly. So currently the population burden of dementia is quite high. There's many millions of people and their caregiving families that are struggling with dementia, and the prevalence is expected to increase in the coming years. And for genetic folks, what do we know about the genetic architecture of dementia, or in this case specifically Alzheimer's disease? There's a small cluster of highly penetrant rare mutations that are associated strongly with early onset Alzheimer's disease. These are relatively rare in the population. And then when we look at the much more common late onset Alzheimer's disease, we see that there's a number of genetic loci that have been identified that have relatively small increases in the effect sizes for onset of this disease. And here's the zoom in of that small panel. There's a couple exceptions we noticed. These triangles out here are two types of alleles you can have in the apolipoprotein E gene that we tend to treat separately. So we look at these polygenic loci together, and then the effect estimate at these apolipoprotein E sites are so strong, we look at them separately. So here's how this looks in a population-based cohort of the health and retirement study. This is the distribution of a polygenic score based on those loci among people who have normal cognition. Here we see that the curve shifts right for people with cognitive impairment, and it shifts right further for people with dementia, as you might expect. And when you do a formal association test, you see that, as folks have noted in this meeting, we have differences in our ability to predict the outcome based on our genetic ancestry and the amount of information we have these scores are trained on. And if we look at the impact of ApoE, we can see the population attributable fraction is quite high, about 20%, and then cumulatively, this polygenic score explains about another 20% of cases. So far I've shown you that there's a genetic risk factors that are associated with our outcome of dementia, and we've been using these population-based approaches. And I'll fill in the bubbles with some other techniques along the way. So our next question was, are there some DNA methylation biomarkers of dementia that we might be able to look at, potentially an intermediate pathway or a biomarker indicator of disease? And we learned from Dr. Bakulski yesterday all about these very useful DNA methylation aging markers. And here we took the residuals of one particular marker and tested for an association with cognition, much in the same way we did with the genetics. But here we're looking at over time. So we've got methylation measured at one time point, and we've got the cognitive outcome measured 2 years later. And here's the density plot of the participants who stayed with normal cognition the whole time. So you can follow this median line where it's going to travel. Right now we see these people tended to have decelerated epigenetic aging at baseline, and they stayed normal cognition over follow-up. Here's the people who transitioned from normal cognition to impairment. You see that median line's moving over. Here's the people who went from impairment to dementia, and the people who jumped from normal cognition to dementia over follow-up. So we're seeing there's some association here with the outcome measured prospectively. And if we try to look at the ability to classify people and make predictions with this, we see there's very, very marginal improvement in our prediction capabilities. We were also curious once this cohort recently released their full complement of methylation data about looking at site-specific measures. I'm interested in what are the genes involved, what are the sites involved. So we performed an epigenome-wide association study looking at cross-sectional association with impairment, cross-sectional dementia, and then over we had to combine together any impairment and any dementia with the incident analysis over time just due to case count. And we were able to see that there are some individual sites that have relatively small effect sizes. We're talking about on the order of magnitude between 3 and 10% methylation differences between these groups. And when we look at them together, we can get illumination of some pathways that might be relevant to us, including things like neuron differentiation and development of different tissues. So here we're starting to build some information about the relationships between potentially some of these DNA methylation as a biomarker tool. We were curious also about if there are modifiable factors for dementia that we can investigate. And the one I'll tell you about first is lead. So what do we know? What's the overall landscape of modifiable factors with dementia? This is a plot from the most recent Lancet commission. They put out every couple years a study of what are all of the modifiable factors for dementia of which they've determined we have causal evidence for, based on the whole body of literature. And I like this diagram. It takes us all the way from early life to midlife factors to later life factors. And you see there's a lot of factors that people in this room are really passionate about. Things like early life education, a lot of these midlife conditions, another social factor, we've got social isolation out here. And they're estimating the proportion that might be modifiable if we're able to intervene on these factors. And this one in here in the late life caught my eye as somebody who's really passionate about environmental chemicals. We see that air pollution is one of these causal factors. Air pollution, particularly the particles that are in air pollution, are complex mixtures of multiple chemicals together. So this premise makes us thinking about what about individual chemicals? What about other, is this a more generalized phenomenon? And what about the temporality or the time course of these exposures? So here was focused on just late life. The US is a global leader in the production of chemicals. This is something we really excel at. Here's a plot of the volume of chemicals produced commercially over time. And here's a diagram. At the time, we were producing over 85,000 different types of chemicals. And at that time, we'd only fully investigated 300 of those for their toxicity screening. This number has improved recently, but there were only 9 that had been fully banned for use due to human health concerns. These were things like asbestos, dioxin, that have very strong negative human health effects. So what this is telling me is there's a lot of chemicals in our environments where we live, where we work, where we play, that have been incompletely characterized for some of these endpoints. And we know that these are really modifiable. So we have a strong history of policies to help intervene and improve the environments for chemicals, which we have evidence here. So one of the things when we want to investigate environmental exposures with a disorder, folks have been thinking about this throughout the day today with social exposures as well. You have to decide on your timing, right? When we're just looking at genetics, we can ignore this axis, but timing is going to be key for this element. So do I want to measure the exposures right when the participant gets their clinical diagnosis of dementia? Sometimes that might be all that's available in our case control studies, but maybe we're curious about exposures that happen a little bit further upstream, maybe when those clinical symptoms start to become apparent, or maybe we even want to go further upstream to be looking, there's evidence that pathology might be occurring earlier, or even in early life might be setting somebody up for a certain course. So we're going to try to investigate exposures during critical developmental periods. This is going to require some creative exposure assessment and study designs because we don't really have the infrastructure for a full life course individual study taking us from the prenatal period up through dementia with serial biospecimens that can allow us to estimate exposures at these time points. So I'm going to try to stitch together a bunch of different projects to bring evidence together. So here's just one recent project where we're looking cross-sectionally among older adults that live in California. And we can see this is plotted by the population density, and my points of lead exposure have disappeared. We've had a little bit of MAC translation issue, but what you would see with these blue dots is that they are the sites of lead-releasing facilities. We have something called the Toxics Release Inventory, where we monitor all the commercial sites, industrial sites that release each type of pollutant in this country, and so we can go back and track them and see how much they've released into the air, the water, the soil. These sites tend to actually be located quite closely to our population centers. So we looked in the Kaiser Permanente population at the proximity of someone's residence to one of these industrial lead releasing facilities to see is it correlated with somebody's cognition at this time point in older adults, measuring on 3 different types of memory. And we saw if we're looking at very tight buffers, so you're living quite close, there was an association with memory. If we're looking at intermediate buffers, it was also associated with memory, and started to attenuate for some subscales when we opened up further buffers, and here was a continuous measure of distance to there. So this is looking at the same time point, so it's in older adulthood. How are we going to stretch into other time points? We can borrow some of these principles that chemicals, we are inhaling them, we're absorbing them, dermally, we're ingesting them, and then once they come in our body, they travel around on the superhighway of our bloodstream, and they're gonna spend time in our tissues with different half-lives based on the properties of that tissue and the property of that chemical. So for example, our bones have very low turnover rates of the hydroxyapatite structure in there. So if I measure lead in your bloodstream, it has a half-life of about 30 days. But if I measure lead in your bones, if it's the spongy trabecular bone of your kneecap, it has a half-life of about 15 years. And the dense cortical bone of your shin has a half-life of about 45 years. So we're playing this role of detectives, of trying to estimate someone's exposure history by comparing their chemical levels across tissues based on our understanding of physiology. Stitching those together across this life course estimation approach. So in addition to those industrial lead releasing facilities, many of us live in housing that was built before 1970, where we can have lead in our house dust and in our water supply. So here we're working with this unique opportunity with the Veterans Affairs Normative Aging Study among participants ages 65 and older. They did one of these lead measures. So here is the device I built during my dissertation. It's a non-invasive X-ray instrument to measure the amount of lead in your kneecap. So here, even though the measure was happening in people in their 60s, we're making an inference or stretching back prior years based on that half-life understanding. And then people were followed up, looked to see when their cognition dropped below a certain level, and we saw that lead in this tissue was associated with higher hazard of developing impairment over that time period. So this is suggesting to us that exposures in midlife might relate to later life dementia. Now these instruments are kind of unwieldy for many reasons I can get into, and there's very few of these in operation. So what we can borrow is our understanding of toxicokinetics, or the rate of movement of these chemicals between different tissues. And Xin Wang, one of our really talented K Award recipients, built a superlearner algorithm to try to predict someone's bone lead based on blood lead and a number of commonly available clinical and metabolic factors. So now we can borrow this into other settings. And we were working with data from the National Health and Nutrition Examination Survey, which is a US population-based survey, exposures at baseline. We've got these estimated bone lead measures, and we were able to connect the data using the remote secure data enclave to incident Medicare claims. So now we've got that temporality that we want. We're able to look at incident AD cases over 25 years of follow-up. We saw very strong associations between tibia lead and incidence or hazard of incident AD over time. So we're starting to build evidence across multiple cohorts, across multiple techniques for measuring exposures that lead might be related here. How can we start to pull omics into this mix? A lot of these cohorts do not currently have, they might have lead, they might have At the time, they didn't have omics, so I'm going to try to pull in now some of our laboratory-based approaches, our experimental models, to investigate some of these omics. So with the epidemiology, I can get to this kind of white box in the middle where I've got evidence that lead's making its way into the body. Something happens in the middle, and we've got AD coming out the other end. Based on our laboratory work, I could show that there's some relationship with AD pathologic pathways I'm not going to get into today because it's not omicsy, but I'm happy to talk about it if you like. But then there's a lot of this non-specific neurotoxicity happening where we have omics measures that are well characterized. So here's just one example where we're working with Rachel Morgan. One of our postdocs is looking at a neuroblastoma cell line. So this is when you start culturing it at day 6, it kind of looks diffuse. And then over time, they start to differentiate, and now they look more stretched out, elongated, like a neuron. So this is under a control, no exposed. Here along the x-axis now are different lead doses, and we can look to see how does that differentiation pattern of the neurons change. Kind of a model of neurodifferentiation that might be happening in kids with lead exposure. And we see that with lead, it's making those neurons have fewer projections, so they're not as likely to talk to their neighboring neurons. We also see that those projections are shorter, so their ability to connect to distant cells is more limited. We also worked with a mouse model of perinatal lead exposure and aged them up to 18 months, which is quite old mouse. Looked at the cortex tissue and performed bulk RNA sequencing on it to see are there gene expression changes that are happening in this brain tissue, things that I wouldn't necessarily have access to in a human study. And we saw all these different genes that are associated with these lead exposure signatures. And then Harang, another student, queried, kind of scraped the internet of all of the human AD postmortem gene expression data to see what are those AD signatures. Clearly these are different, right? We've got the lead measured in the mice, we've got the AD measured in the humans, but we're testing to see are there overlaps of these genes over these pathways. Can we start to make some inferences about the cell types involved and the genes involved across these agents. We just very recently were able to start generating some metabolomics data across these same mice, so we'll be able to compare some of the downstream metabolites that are here in both the cortex, so the same tissue that was in that RNA expression paper, and plasma, hopefully with some inferences for our human study that has more metabolomics data available. So we're starting to pull it together, some of our evidence in bulk tissues. What does this idea of cell type matter in omics tissues? And this has been a major topic that a lot of folks at this meeting have been curious about and talking about, and why does cell type matter in our non-genetic omics studies? So here's one example where I came in with a hypothesis about a specific cell type that I thought was involved, these cortical neurons. We were looking at that lead exposure, perinatal lead exposure model, working with some of the cortex tissue at 10 months at that time, and we sorted out just the neurons. I used fluorescence-assisted cell sorting to pull just the neurons and ran some DNA methylation measurements around there. And we found kind of a modest number of associations where we have this dose-dependent relationship between lead exposure and DNA methylation. But we started to get curious about, am I too narrow in my focus? Should I be looking at other cell types? Should I be looking at other brain regions? And this is relevant because our cells are differentiated by epigenetic patterns. This is the classic Waddington diagram of, up at the top, we've got our undifferentiated stem cells that are toady or pluripotent. They can go and become anything they dream of, whether it's muscle cells, liver cells, nerve cells. These cells have different shapes. They have different forms, different function. And a lot of their differentiation state is locked in by epigenetic factors, including DNA methylation. And this is a complication for us in population health research because we're often grabbing people's blood, we're grabbing saliva, and while to the naked eye, this looks quite homogeneous, if we zoom in, it's quite complex. So there's a lot of different types of cells in there, and each one of those cells has a different epigenetic underlying pattern. So for example, here on the y-axis, I've got DNA methylation at 3 options, because any individual cell only has 3 options. It can either have 100% methylation if both copies are methylated of that position. It can have 50% methylation if one's methylated and the other's not, or it can have 0% methylation if neither copy is methylated. what we're measuring in our studies most of the time in human epi is a per person mean. So we're just getting an average across all of these. And that's how we can get some intermediate values that are not so discrete at 100, 50, and 0. What's the relevance here? So here's that same picture where now I just have multiple cells in each of those 3 cartoon types. But if I have an exposure, in my case lead, or in your case might be a social factor, let's imagine a scenario where our exposure is changing the methylation. Here I'm showing an increase of methylation, but you could just as easily show this with decreasing methylation. There's different biologic scenarios that could be happening there. We could have a direct effect, which would be every cell in the tissue is responding to the extent that they're able. We could have a vulnerable cell type. So this was my original hypothesis. I think that the neurons are going to be most sensitive. The other cells might be staying the same, and then that vulnerable cell type is a high responder. Or we might imagine a scenario where there's no methylation changes at all. All of the cells' methylation states are staying exactly the same, but what we have is a shift in the proportion of the underlying cell types that are present. You might imagine that there's an inflammatory status happening, so the immune cells are making their way into the tissue. Maybe you're having fibrosis, so we're getting fibroblasts in the tissue instead of the other specialized cells. All 3 of these scenarios, I think, are strongly biologically relevant, but our ability to intervene would be quite different in each of these scenarios. The types of strategies we might take to support somebody experiencing changes in these ways might vary. So I think it's helpful to try to figure out which one of these is going on in your study, and thankfully there's some strategies that can help us to do that these days. One way that I was excited to work with this was in a single-cell RNA sequencing framework. We were working with that same lead model. You can tell I'm very good at scavenging tissues that are around in the department on the floor upstairs of me, so thank you, Dana, Joel, and I, for being very generous with all of these tissues. Here I was working with hippocampus tissue, which is a key learning and memory region in the brain implicated in Alzheimer's disease. And this time when I dissociated those cells into a single cell suspension, I didn't just select out the neurons. Now I fed all of the cells into a microfluidics device through the 10x Genomics platform. So each cell starts going through the tube, and it gets linked up into a little oil droplet that has barcodes in it. So then when you lyse the cell, all of the RNA transcripts get labeled, and you can go back and tell which cell they came from. So now we go on and sequence those barcoded stretches, and we know what cell our observations come from. And our data structure is a little bit different. We may be used to having a data set where each column is a sample, and each row is a gene. But here now we have, for one sample, we might have up to 5,000 columns, because each cell gets its own column. And then we've got multiple samples. Okay, so the data structure is a little bit different than a bulk tissue experiment. And here's what the output, or one way of visualizing the output. So now each dot is a cell that's been sequenced, and it's painted by how similar those cells are, clustering them, grouping them into cells which are more similar to each other. And you can look at what are the marker genes, the genes that are known to be upregulated in those tissues. For example, microglia have this gene turned on, oligodendrocytes have this gene turned on, endothelial cells, and so forth. And you can identify which one of these cell clusters are predicted to be each one of these cell types, and then we can estimate what's the relative proportion of each cell type that's present in our sample. Here I'm going to show you this same exact plot. Right now it's painted by the cell cluster identity, and the next plot now it's painted by your treatment group, whether it came from a control mouse or a lead mouse. And if you notice, like if you just squint, they're not directly on top of each other. It seems to be some shifts in those. And if we just look at those proportions, we see that there's a couple cell types that are higher in lead. And so these oligodendrocytes appear to be increased in lead. I don't know if that's a upregulation of those or a loss of others, because I've only got this one exact time point. But these are these glial or support cells that wrap around the axons and help those neurons signal faster. So what this tells me is that I was thinking too narrowly at one cell type, and this is helping me think more expansively about some of these support cells that might be vulnerable. With this data type, we can do a traditional gene expression analysis, look to see are there differences in gene expression with our exposure, and we see some genes that we expect a good positive control check that should be associated with this chemical. They are enriched in a lot of the Alzheimer's pathology genes that we pulled previously. We also see that there's a lot of cluster-specific responses. So it seems like there's some uniqueness in terms of what these different cell types are choosing to respond to lead in. Here's the genes that overlap across cell types, but there's a lot of genes that are individual cell types are differentially regulated. So for example, our microglia are really turned on the protein folding pathway, and some of these different cell types have different pathways that are either up or down regulated. So in this case, it looks like with this one experiment, we're seeing all three types happening here, where we've got some responses that seem to be generalizable across cell types, some that seem to be specific to a cell type, and some that seem to just be changes in the cell proportion. So I show this as an illustrative example, but when we do this type with deconvolution in our human studies, there's also widespread differences in these types of responses as well. You may have noticed in our single cell experiment that I had a really small proportion of neurons captured. I think the neurons were very upset with me trying to dissociate them in a single cell suspension. So we've been experimenting with other techniques that allow us to keep those cells in the context of their histopathology. So some of these more spatial techniques where we don't need to disrupt them so much. But these have created a whole new fun data analysis challenges that I get to learn about and enjoy. So here we've seen that LEDs working across multiple different omics levels, and sometimes in a cell type specific manner. Is this something specific about lead, or are there other environmental chemicals that might be working the same way? So here we looked at a related metal, cadmium, that may have some similar associations. I'll go through this relatively quickly. So we are often exposed to cadmium in our daily lives through our dietary factors and through cigarette smoke exposure. Some plants are preferentially very good at sequestering cadmium from the soil, and then it enters our body through those routes. Here we were looking, again, at data from the National Health and Nutrition Examination Survey, where they measured cadmium in blood, which has a relatively short half-life, so it's reflecting recent exposure to cadmium. We linked it to public mortality data to see if somebody died due to Alzheimer's disease. And we saw that blood cadmium was associated with increased hazard of dying due to Alzheimer's disease. But if we looked at another biomarker, if we looked at urinary cadmium, our kidneys are especially good at sequestering cadmium. So the half-life in urinary cadmium is quite long, 10 to 15 years. And we see that that has an even stronger association with the outcome. So again, trying to piece together some of these exposure windows by using different exposure biomarkers. My new colleague, Hao, who just started at our school, had run a cadmium mouse model. Here he was working with kind of adolescent or young adult cadmium exposures, aged them up to about 8 months, and he showed memory deficits in these mice. He also showed these interesting depletion of adult neurogenesis with cadmium exposure in these mice. So there's some behavioral as well as pathologic evidence is as a foundation, and thankfully for me, he had some cortex tissue that was present in the freezer that he was willing to let me do some omics on. And we tried out another measurement technique that I think might be a future application for human population-based studies as the price for this comes down, and I'll show you some of the advantages to this. This is a direct sequencing method. So a lot of us, when we use the Illumina arrays, there's a prior step of pre-processing in the lab where we do bisulfite treatment. And basically what that does is it ends up collapsing all of the DNA modifications into one. So the DNA methylation, the hydroxymethylation, the formylation, the carboxylation are all smushed into that measure, and we call it DNA methylation, but really it's a mixture of all of those modifications. This technique has the advantage that we can resolve those different types of modifications. So we don't do that, there's no prior bisulfite treatment, we just feed the DNA directly into this pore. There's an electrical current that's measuring a signal from each base, and we can identify the genetic sequence of this. And there's base calling algorithms where we can identify a whole bunch of different epigenetic modifications, including the cytosine ones I just mentioned, but also some adenosine modifications. There's all kinds of modifications on the genome. So we're looking at reads that are over 7,000 base pairs long, and there were over 13 million CpG sites that we were able to capture a sufficient coverage. So we're able to capture both what we call traditional DNA methylation, or this 5-methylcytosine, as well as hydroxymethylcytosine, which is the same thing, it just has an OH group stacked onto it. And in the brain of these cadmium mice, in the control mice, there was approximately, of all CpGs, about 60% of them had DNA methylation and approximately 15% hydroxymethylation. So this is a relatively abundant marker that we're normally smooshing together with the DNA methylation. And physiologically, the hydroxy is supposed to be on the pathway to demethylation. So they're working in opposite directions. So here's what those results look like from those cadmium-exposed cortex. If we look just across the genome, where are these modifications happening? We see that there's not a lot of modifications at the CpG islands, but if we look at the open C regions, there's a lot of methylation. There's also a lot of hydroxymethylation. Same thing with the shelf. And the differential methylation and hydroxymethylation with cadmium exposure was occurring at some of these gene pathways that were relevant for neurologic outcomes. And here's what the volcano plot looks like. We can see that cadmium is kind of causing this hypomethylation, so we're losing methylation. It's also causing a hydroxymethylation. So these two are going in opposite directions, and if we were using traditional approaches, they would cancel each other out. And here, for example, is the same gene on both sides. So this gene is associated with lower methylation with cadmium, but higher hydroxymethylation. And what does that look like? Here's a differentially methylated region. On the x-axis is the genomic loci. The y-axis is the amount of DNA methylation, and in pink are the controls. They've got lower methylation, and the cadmium exposed have high methylation. That same region with hydroxy is exactly the opposite, where we've got the exposed have low methylation and the controls have high hydroxymethylation. So they're complete opposite responses that under our normal, like an Illumina methylation array would have collapsed all of these together and you would have seen no effect. So I think there's a lot of promise with this technique in terms of being able to resolve with greater precision what are some of these epigenetic signals in our population-based studies. And the NIA, I'm grateful, just started something they're calling the ToxAD Consortium, where they're gonna allow us to look at some of these chemicals we've been investigating with a polygenic mouse strain. So this will bring together both of those areas where we've seen AD is very polygenic and be able to look at those chemical exposures in that context. Here I've been kind of cherry picking chemicals that you want to start in an area that you think you're going to see a strong effect, but can we look more widely to investigate the chemical exposome? So here we did what's called an exposure-wide association study of cognition, where we were looking at people in the National Health and Nutrition Examination Survey. We're looking at over 100 chemicals, and we just cross-sectionally looked at cognitive status among persons ages 60 and above. And we saw that there are some chemicals here are two major pesticides that are associated with lower cognitive status. And then this is a more broad phenotype. So here are those same two pesticides I just showed you. Here's cadmium, here's lead, and there's a whole bunch of other chemicals in between that we can now go and pursue and investigate. Nguyen showed recently that there's very strong racial, gender, all kinds of disparities in the chemical exposures of what people are observed in people's blood and people's urine. So here showing chemicals that are elevated in black women relative to white women, and here are chemicals elevated in white women relative to black women. Here's lead we see as an important disparity chemical. And we know, those of you know very well as social scientists that exposure patterns are socially constructed and variable. Here's some data on the distribution of lead levels across the country. And Tara Jensen, one of our trainees, was curious about, do these chemical exposures mediate or perpetuate some of these social responses? So is the disparity, racial disparity in cognition observed in this population mediated through those lead levels? And they found a small percentage was. So this gives us an opportunity to think about interventions. What kind of actions could we do to reduce some of these social harms? Maybe one of them might be through targeting some of these neurotoxic chemical exposure pathways. In these last couple slides, I want to show you some additional ways that we were trying to link social context with chemical screening. And here, I'm relying heavily on partnerships of people and your colleagues who have expertise in these type of social measures. So Laura Arboleda Marina, one of our doctoral students whose diversity supplement was unfortunately just canceled, but we are regrouping and finding another way to move this project forward, was curious about constructing a social vulnerability index, so an aggregate of multiple social factors together, seeing that in this population, there's very strong association with cognition. So people have lower cognition, the more socially vulnerable they are. And we wanted to look to see, can we combine our investigation of chemicals and social factors together? So here I'll show you one chemical before all of them. Here's a chemical called diethyl thiophosphate. It's a metabolite of organophosphorus pesticides. You might be familiar with like chlorpyrifos, would be the parent compound. And here are the people who have high social vulnerability, so our most marginalized people. We have the steepest slope, so higher chemicals with lower cognition. Here are people with medium social vulnerability, so there's some ability to buffer those chemical effects, and then here are people with the lowest vulnerability, so the most advantaged folks have the most flat slope here. And here's when we look at it across all chemicals, we see there's a number of new chemicals that pop up once we're considering social context. We never would have identified these in the original screening, but now by bringing together our chemical and our non-chemical stressors, we're able to get a more full picture of what people are experiencing. So hopefully together I've shown you some examples about how we can take what we've learned about chemical exposures. We can look at the pathology directly that we think are involved in some of these specific disorders. We can also do some of these more broad omics type investigations of intermediate steps. And depending on what we find here, these are opportunities for intervention that might vary based on the types of factors that we identify. So my key takeaways, I focused on dementia, but hopefully some of these principles are useful to you in other areas. This idea that integrating both the social, the chemical, and genetic factors is a really crucial way to get at prevention and intervention opportunities. Some of the complexities with all of this, we learned that timing really matters in terms of windows of susceptibility to these social and chemical exposures. We saw that combining multiple layers of omics can be useful. We learned different bits of information across each of those layers, and I think this is the biostatisticians have been asking what are the future opportunities. This is certainly one of those elements. We also saw that some of our cell types can have common responses across cell types, but there might also be specific responses to those exposures. And then lastly, trying to bring together some triangulating of evidence across study designs, some collaboration can help us overcome the shortcomings that any one of these approaches might have individually. And I'll just leave you on this picture I took this summer at the Detroit River International Wildlife Refuge. It was a site of a Chrysler automotive plant until 1990, and then an industrial brownfield until 2001, and was designated a Superfund area. So that's our country's designation for the most polluted sites we have. And it's now a spectacular sanctuary for unique wildlife, and it's an amazing bird migration time for it right now. So it reminds me that our environments are highly modifiable. We know the levers to press on with respect to policy and intervention to help make some of these changes. And so I think these types of research that people in this room are doing are very influential in terms of prioritizing exposures for prevention, and the genetic side is helping us target mechanisms that might be useful for intervention. So I want to thank you for your attention today and say I've really enjoyed this meeting.
Hi, Kelly, thanks for the presentation. It's great. I mean, I personally learned a lot from it. So I have a question about the cell types you presented. You presented basically the three different models, the direct effect, cell, like vulnerable cell types and the cell heterogeneity. And your study shows evidence for all the three, but you also mentioned that they have different like implications in terms of prevention, intervention. So, I'm wondering, like, so in ideal scenario, say, what if you say only have evidence for direct effect, or only evidence for the vulnerable cell or cell heterogeneity? What are the kind of differences in the implication in terms of the relationship between the exposure and outcome? Maybe also for like policy implication in terms of intervention, prevention. So.
Oh, that's a great point. Thank you. Yeah. So I might think about if we're seeing actual direct methylation changes, and I also showed gene expression changes that are directly happening, and those cells are changing their expression profile. Then we might move to some of the folks in the room have been talking about druggable targets, where we're trying to either bring up or down that gene expression in that cell type. Our ability for specificity around those are still improving, at least on the methylation side, but there you might want to go after that specific gene, or you might want to go after that specific protein, for example. If we're seeing changes in cell composition, that might be something differently entirely. So if you're seeing, for example, an expansion of inflammatory cells, then maybe we want to dampen that immune response. That might introduce an opportunity for a different suite of druggable targets that are helping up or down regulate for example, the neutrophil infusion into that tissue. So I think for me, there's opportunities if we're trying to change the cells that are there, or if we're trying to change the behavior that the cells that are already there are undergoing in those cases. Yeah, both I think have a lot of promise and opportunity. Yeah.
Thank you, Kelly. That was fantastic. I really appreciate how you're bringing together these laboratory approaches with population data, and I just think that's really fantastic. I have a lot of questions, but a couple. First, because you were talking about how the Illumina array kind of collapses all these different types of methylation into just DNA methylation, and how that might mean we're missing out on some of the impacts of these exposures. What do you think the implication of that is for the epigenetic clocks?
Oh.
Like if we're not seeing, for example, associations between biological aging and certain exposures, could it be that it's because of what you were showing? Is that something? And then the other question I had is, how do all these exposures compare to smoking? And just, that's something that we've studied a lot as economists and social scientists, and I'm just curious, all these, things that we're exposed to in the environment, how they compare to smoking.
Yeah, those are great points. Thank you so much, Lauren. I'll tackle the second one first. Yeah, cigarette smoke exposure is a complex mixture. There's over 1,000 chemicals in a cigarette, and when you oxidize it, we get even more different types of chemical products. So certainly when I was performing that exposure-wide association study, at least 25 of those chemicals are known cigarette smoke byproducts. So the way in which we handle this are we are adjusting for people's categorical smoke exposure. We're also adjusting for something called cotinine, which is a biomarker of smoking, and we're also adjusting for pack years, so a cumulative measure. So we've got 3 different smoking adjustment factors in there, particularly for those ones that are known metabolites of cigarette exposure. So you're absolutely right. We want to be very cautious and make sure that we're not over attributing something to cigarette smoke exposure, that we're dialing in on the specific chemical. Those chemicals are often not specific to cigarette smoke. We can be exposed to them through other routes. For example, I love to eat barbecue food, but that combustion can also occur through some of those pathways as well, or air pollution contains some of these same chemicals. So that's an interesting element. And then you were talking about the different modifications. Yeah, there's a lot of interest in this, and there's even interest right now in the environmental space. People are into RNA modifications. So if you have RNA sequencing data, there's modifications on there, like methylation on the adenosine molecule, for example. So I think this is going to be another kind of future element that traditionally we weren't able to resolve those factors, and it's only going to get better and better, our ability to distinguish those. I don't know the relationship between the clock and the different factors. Although I would be curious to check in with Dr. Belsky about that. We've got some great future ideas for things to look at.
I had a different question, actually, but I'm going to switch it because I thought the smoking thing was interesting. I thought what you were asking was, at least the way I was framing it in my brain was could you use smoking, because so many people are familiar with it, as a unit of measurement of this exposure is 5 times as bad as smoking a pack a day or something like that. That's something that people relate to intuitively. And so could you comment on that? And maybe, I guess my original question was also about, you mentioned route of exposures. Are there general principles you've learned from all of this work when you think about
the route through which you're exposed, whether it's your drinking water or air or in the food supply or some other means that are particularly problematic.
Yeah.
So sorry, that's kind of two questions.
No,
but
this is great. Yeah, for in terms of cigarette smoke exposure, because yes, we always try to like benchmark it to something people are familiar with, and I forgot to do that here. Usually for cognitive stuff, I benchmark it to aging. So I'll say like the effect of this exposure is the equivalent of like 1 year increase in age, something like that. The relationship between cigarette smoke exposure and dementia is really complicated from an epidemiologic perspective. We have so much selection bias that sometimes cigarette smoking among older adults can look protective for Alzheimer's disease. So I try to avoid that one just for this outcome, but I agree in other outcomes that would be a very valuable public communication tool. It's just that my relationship between cigarette smoke exposure and dementia is kind of fraught in this particular analysis, but I'm really excited about making sure that we're communicating this in a way, because knowing what a 1 unit increase in diethyl thiophosphate is not that helpful in terms of people don't know what magnitude they're around. The second question was related to route of exposure is very interesting, and our susceptibility to different routes of exposure varies over time. So for example, like if me and my little niece Evelyn, who's only a year, ingest the same amount of lead in the drinking water, much more is going to cross her intestinal permeability than mine. So there's age-related differences in our barriers to these. Our blood-brain barrier and our gut barriers establish further postnatally. There's also differences in, for example, some of these gases and air pollution are heavier. So Evelyn's gonna have a higher exposure level because she's down by the floor, and she also has a higher respiratory rate. So for many different reasons, there's different vulnerable populations for these different exposures. People who are pregnant are mobilizing a lot of their bones for constructing a skeleton of a fetus. So that actually, their historic lead can become an endogenous source of lead because they're mobilizing all of that archived lead that's in their bodies. We see that also with our dementia folks. Their blood lead levels, if I'm measuring them at the ADRC, it might not be exogenous lead, it might be their osteoporosis is mobilizing that lead right there. So there's, yes, many issues, not issues, but specificity we need to take in terms of who is most vulnerable to different toxicants, and it's gonna matter based on their age, their body size, you know, it's gonna matter based on their health status, their nutritional status. If I am eating a diet that's rich in iron, then the lead is not going to be able to bind the divalent metal transporter, so less of it's gonna cross my gut barrier as well. So we've got interaction with a lot of different factors, which I view social exposure as kind of this overarching element that is an indicator of a lot of these specific pathways that are coming underneath it.
Thank you so much. Wow. Let's thank Dr. Bakulski here for our excellent end to the conference. So the reason I'm stopping it for a minute is that we have a set of people who are leaving for the airport and wanted to invite us to take a breath, but also come up and talk, and ask further questions now that we're officially done. And I also wanted, as we're leaving, for you to recognize Mindy and Tommy and Elliot as you walk out, because they're the ones who put this all together. I think this has been a great space. Everything's worked out really well. It's been very invigorating. So, but tell them that as we leave. So, with that said, we're officially adjourned. But keep coming up and asking Kelly and others about the work that they presented today. Thanks very much for coming.
Thank you.